Doesn't really look that great, huh? Whatever. At least it's there, I guess. All right, let's I think let's review enzyme structures and functions. Then do enzyme kinetics, and then Anki, and then move past and do okay. So I think if we do review, let me see if this will work, because we have to get it done by Saturday, so Sunday I can at least have that time to do the other stuff, and have a little bit of of a habit for the Saturday and Sunday. So we'll be review enzyme structures and function. Take enzyme kinetics. Take enzyme kinetics. Then we'll do Anki review. We'll add enzyme kinetic cards to each deck that I can, and then we'll do. Well, yeah, Anki review. I did that. Okay, and then we'll do DNA and gene control. DNA and gene control. And then for Friday, that'll leave us with 1, 2, 3. And Saturday, 1, 2, 3. And we finish. The Sunday, we'll do what's the timing charts and do the passage. That sounds like a good plan, jolly boy. I woke up late today. Won't happen again. But we'll go 9 to 9. All right. Before we start, let me clear up some things. Darwood Sandstorm. Yeah, I got all these dishes. I just gotta have a clean area. Gotta have a clean area, clean mind. All these dishes don't look good. I'll be back. All right. Let's also include. I want to take a cold shower once it gets warm. Maybe tonight or something. And I need to do the documents for the Air Force for the security clearance. Submit documents for the Air Force security clearance. Ooh. Let's also see if we can do a little workout. Maybe we can do 2 dumbbells and push-ups. Okay. All right, let's plan. We gotta get started. We're late.
to you about enzymes and how they're critically important pieces of cellular machinery. But first, let's review the idea that biochemical reactions happen in the body all the time. Almost every cellular process involves a biochemical reaction at one point or another. The TCA cycle is actually just a series of different.
Oh man, I just realized you guys don't know what I'm doing. There you go.
Different biochemical reactions in carbon metabolism, DNA replication.
Oh no, that's just me. Oh shoot, we gotta read the high yield. I forgot. We can't do on-cube until we read the high yield. I gotta write that down. Word high yield guide and then on-cube review. Ooh, we got some work ahead of us.
Which needs to happen before a cell can go through mitosis is also just a series of at one point or another. The TCA cycle is actually just a series of different biochemical reactions in carbon metabolism. DNA replication, which needs to happen before a cell can go through mitosis, is also just a series of reactions. And this also applies to the expression of genes, going from DNA to RNA to protein. And we need enzymes because enzymes make all of these reactions go much faster. And let's look at this idea a little more deeply and how a reaction will go on differently when it has an enzyme versus not having one at all. So you may be familiar with the reaction where water and carbon dioxide can combine to form carbonic acid. And this is a reversible reaction, so it can go backwards and forwards. Now, when people make soda or any carbonated beverage, they'll start by pumping that soda can full of CO2. And while some of that CO2 will dissolve in water in the can, the soda making companies are able to get a lot more CO2 in the water by using this reaction. The abundant CO2 will react with the water to form some carbonic acid in the can. And when you go to open the can, you'll hear a pop sound, which is really just a bunch of CO2 escaping. But after that, the soda will start to fizz really slowly. And what's happening here is the carbonic acid that was made before is slowly dissociating back to carbon dioxide and water as CO2 escapes. And that extra CO2 that's being made will come out of the soda solution, and you'll see it as little bubbles floating around. But what happens if we then take this person over here and he'll pick up a can of soda and take a drink? That person might notice the soda will start fizzing a lot more once it hits his or her tongue. And this is because humans have an enzyme in their blood and saliva called carbonic anhydrase. And this makes the carbonic acid turn into carbon dioxide and water much more quickly. So more CO2 will come out of the can and it'll fizz more. And this is just one of the many examples of how enzymes make reactions go faster. So how exactly do the enzymes make the reactions go faster, though? Well, they use a bunch of different catalytic strategies to push reactions along a little more quickly. And I'm going to talk about a few of those strategies just to give you an idea of what enzymes are doing. So first I'll mention acid-base catalysis, which happens when enzymes act like either acids or bases. Now remember that acids and bases are proton donors and acceptors. And if you look at this type of reaction, which if you remember from organic chemistry is a keto-enol tautomerization reaction, we have a proton moving from a carbon atom to an oxygen atom. And since acids and bases are pretty good proton carriers, they could both help with this reaction and make it go a little more quickly by helping to move that proton around, instead of this molecule doing it by itself. Our next catalytic strategy is covalent catalysis, which happens when enzymes form a covalent bond with another molecule, usually their target molecule. Remember that covalent bonds involve two molecules sharing electrons. And looking at this reaction here, we have a decarboxylate. And since acids and bases are pretty good proton carriers, they could both help with this reaction doing it, which happens when enzymes form a covalent bond with another molecule, usually their target molecule. Remember that covalent bonds involve two molecules sharing electrons. And looking at this reaction here, we have a decarboxylation reaction going on, which, if you remember from organic chemistry, is when a carboxy or CO2 group is being taken off a molecule. And if you remember, these reactions usually have a lot of electrons moving around. So if we had a covalently bound enzyme that could hold on to some electrons, be an electron carrier, or what some people like to call an electron sink, then that would definitely help this type of reaction move a little more quickly. Next, we have remember that to move that proton bond is when a these bound, what people like to call an electron sink, then that would definitely help this type of reaction move a little more quickly. Next, we have electrostatic catalysis. Now, if you remember, DNA is a very negatively charged polymer because of all the negatively charged phosphate groups that we find in DNA. So if an enzyme had a metal cation on it, like magnesium, we could use it to stabilize the negative charge found in DNA and make it a little easier to work with. And DNA polymerase, which is the enzyme that allows DNA replication to occur, does exactly this. And in order for it to help with DNA replication, it needs to find a way to counteract all of the negative charge on DNA, and magnesium ions totally come in handy there. So the last catalytic strategy I want to mention is a little more general, and it has to do with proximity and orientation effects. Remember that in order for two molecules to react with each other, which is usually what enzymes help out with, they need to physically collide at some point. If we have molecule A and molecule B, they'll only react once they crash into each other. And a lot of enzymes are able to bring two molecules close together so that these types of collisions happen more often, making the two molecules react more quickly. Also remember that the orientation of the two colliding molecules in space is also really important. If molecule A and molecule B collide, but one of them is upside down or not in the correct position, then the collision may not result in a successful reaction. So enzymes also make sure that the two molecules will collide in the right orientation. And all of this increases the frequency of collision in general, but also helps to make sure that those collisions are successful and result in a reaction. So what did we learn? Well, first we learned that the role enzymes play is to make biochemical reactions happen more quickly. And the next thing we talked about were 4 of the many different catalytic strategies that enzymes can use. We talked about acid-base catalysis, which helps with proton transfer. We talked about covalent catalysis, which helps with electron transfer. We mentioned electrostatic catalysis, which deals with stabilizing charge. And finally, proximity and orientation effects, which increase the frequency of successful collisions between molecules that we want reacting together. Two molecule collision effort. Well, first we learned that the role ends in the right orientation. So what did we? Those collisions are successful. The result of play is to make biochemical reactions happen more quickly. And the next thing we talked about were 4 of the many different catalytic strategies that enzymes can use. We talked about acid-base catalysis, which helps with proton transfer. We talked about covalent catalysis, which helps with electron transfer. We mentioned electrostatic catalysis, which deals with stabilizing charge. And finally, proximity and orientation effects, which increase the frequency of successful collisions between molecules that we want reacting together.
Sure. Real quick, let me just. Okay.
Today we're going to talk about how enzymes can influence a reaction's activation energy. But first, let's review the idea that enzymes make biochemical reactions go faster. And in order to do that, they use a bunch of different catalytic strategies. Now, there are lots of different catalytic strategies that enzymes use, but a couple of the key ones are acid-base catalysis, where enzymes use their acidic or basic properties to make reactions go faster by helping out with proton transfer. There's also covalent catalysis, where enzymes covalently bind a reacting molecule to help with electron transfer. There's electrostatic catalysis, where enzymes use charged molecules or metal ions to stabilize big positive or negative charges. And we also have proximity and orientation effects, where enzymes make collisions between reacting molecules happen a little more often. So, what effect do these catalytic strategies actually have on a reaction? Well, let's look at a sample reaction where we're having molecule A being converted to molecule B. Now, we can look at the process of this reaction using something called a reaction coordinate diagram. And here we'll plot the energy state of our molecules against the progress of the reaction. So essentially, using this graph, we'll follow the energy level of molecule A as it's converted to molecule B. Remember, the molecule's energy level is related to its stability, and something that is a lower energy state is more stable, and for something to transform to a more unstable form, it needs an input of energy to get there. So looking at this graph, you'll notice that the energy of molecule A will rise up pretty high and then drop all the way down to the energy of molecule B. Talk about how enzymes can influence a reaction's activation energy. But first, let's review the idea that enzymes make biochemical reactions go faster. And in order to do that, they use a bunch of different catalytic strategies. Now, there are lots of different catalytic strategies that enzymes use, but a couple of the key ones are acid-base catalysis, where enzymes use their acidic or basic properties to make reactions go faster by helping out with proton transfer. There's also covalent catalysis, where enzymes covalently bind a reacting molecule to help with electron transfer. There's electrostatic catalysis, where enzymes use charged molecules or metal ions to stabilize big positive or negative charges. And we also have proximity and orientation effects, where enzymes make collisions between reacting molecules happen a little more often. So, what effect do these catalytic strategies actually have on a reaction? Well, let's look at a sample reaction where we're having molecule A being converted to molecule B. Now, we can look at the process of this reaction using something called a reaction coordinate diagram. And here we'll plot the energy state of our molecules against the progress of the reaction. So essentially, using this graph, we'll follow the energy level of molecule A as it's converted to molecule B. Remember, the molecule's energy level is related to its stability, and something that is a lower energy state is more stable, and for something to transform to a more unstable form, it needs an input of energy to get there. So looking at this graph, you'll notice that the energy of molecule A will rise up pretty high and then drop all the way down to the energy of molecule B. And we can actually define a couple of values from this graph. The transition state of a reaction, which is represented by this double dagger symbol, is the highest energy point on the path from A to B, and it's where you'll find the most instability throughout the entire reaction. Now, the difference between the energy level where we start and the top of our graph at our transition state is what we call the delta G double dagger, or the free energy of activation. And this is the amount of energy that A needs to have in order to break the reaction barrier to ultimately get to point B. You'll also notice that there is a difference in energy between point A and point B, and we call this the standard free energy change for the entire reaction, and it represents the net change in energy levels between our reactant and our product, and it's also the energy that is released into the environment once the reaction is over. Reactions you typically look at will have their products at a lower energy state than their reactants, since that makes the reaction spontaneous. Now, it's important to recognize that it is the free energy of activation energy value, which is the difference between point A and the transition state, that usually determines how quickly a reaction will go. And usually this energy value is much higher than the free energy change for the reaction, which is why enzymes speed up a reaction by lowering a reaction's activation energy. Now, I want to quickly point out that you may see delta G double dagger written out as EA in some textbooks, and you may see the standard free energy change for the reaction written out as E reaction. And I'm just letting you know that you might see both sets of terms used from time to time. Now, let's look at an analogy to get a closer look at how this all works. And let's say there's a giant hill that you're trying to climb, and it's a pretty steep hill that goes up really high. but you need to get to the other side of the hill. Now, this would be a pretty scary thing on its own, since you would need to go all the way up and then all the way down the mountain to get to the finish line. But if I were to give you a shovel, then now you could dig your way through the mountain and not have to climb up so high. In this example, the shovel represents an enzyme, and the hill represents the activation energy barrier that prevents you from getting to start to finish. By using the shovel, you're able to lower the height of the hill you have to climb. But in both cases, it's important to recognize that you still started and finished at the same points. So let's go back to our example from before with our reaction coordinate diagram. But now, let's say that the reaction has a catalyst. So with the catalyst, the activation energy barrier that molecule A has to overcome in order to get to point B is much smaller. And this will mean that your reaction will have a transition state with a much lower energy, meaning that it's more stable with the enzyme, and also that the reaction as a whole. Now, it's really important to recognize that, like our example where you're trying to climb the hill, finish line. But if I were to
This is going actually. Oh, that's right. That's right. Anyways, let's check. This one.
to give you a shovel, then now you could dig your way through the...
I didn't complete this before the 21st. Let me write that down too. I gotta work on that tonight. And also send the other one. Yeah. Alright, let's continue. Sorry about that.
The mountain and not have to climb up so high. In this example, the shovel represents an enzyme, and the hill represents the activation energy barrier that prevents you from getting to start to finish. By using the shovel, you're able to lower the height of the hill you have to climb. But in both cases, it's important to recognize that you still started and finished at the same points. So let's go back to our example from before with our reaction coordinate diagram. But now, let's say that the reaction has a catalyst. So with the catalyst, the activation energy barrier that molecule A has to overcome in order to get to point B is much smaller. And this will mean that your reaction will have a transition state with a much lower energy, meaning that it's more stable with the enzyme, and also that the reaction as a whole will have a much lower activation energy. Now, it's really important to recognize that, like our example where you're trying to climb the hill, the enzyme will not be changing the starting and ending points of the reaction. It doesn't change molecule A or molecule B. Your starting and ending points are always the same, and the only thing that changes is the path that you take to get from A to B. Now, since our starting and ending points aren't changing, it follows that the enzymes are not used up when they catalyze a reaction, and there is no permanent change to the enzyme following a reaction. So what did we learn? Well, first we learned that enzymes work by lowering the free energy of activation of a reaction, making it much easier for the reactants to transition and form products. And we also learned that the free energy of the reaction doesn't really change when you use an enzyme and when you don't. Second, we learned that despite the change in pathway to get from A to B, the reactants and products do not change when using an enzyme versus when not using an enzyme. And finally, we learned that enzymes are not consumed when they catalyze a reaction, and the same enzyme can catalyze reactions over and over again. So today I'm going to talk to you about the induced fit model of enzyme catalysis, and how this concept can tell us a lot about how enzymes work. But before we do that, let's review the idea that enzymes make reactions go faster. And when you look at a reaction on a reaction coordinate diagram, you'd see that the catalyzed reaction would have a much smaller activation energy than the uncatalyzed one. Also remember that because of this, the energy of the catalyzed reaction's transition state is far lower than the energy of the uncatalyzed reaction's transition state. So what do enzymes look like? Well, most enzymes are proteins, or at least partially made up of protein, and substrates are any molecule that an enzyme will act on. And often, these substrates are the reactants that the enzyme will ultimately help turn into products through a reaction. Now, enzymes also have what is called the active site, which is the location on the enzyme where substrates bind, and that's where the reaction ultimately happens. And it's important to recognize that every enzyme has a unique active site that will only bind to certain substrates. And just to clarify, I've referred to the active site here as both of the notches found on the enzyme, and not the space in between them. So here, of the two substrates I've drawn, the enzyme will only be able to bind to substrate 1, since they fit together like puzzle pieces, whereas the shape of substrate 2 isn't going to fit nicely in the enzyme's active site. Now, since enzymes have unique active sites, we say that enzymes are specific to certain substrates, and by extension, certain reactions. But let's dive a little deeper into what happens when enzymes and substrates bind each other, and how that binding pattern changes as a reaction progresses. So first, you'll have your enzyme here and your substrate over here, and I'm just going to label this with a number 1, since it'll be the first thing that happens in the sequence of events to come. And at this stage, nothing has happened yet, and the enzyme and the substrate have yet to come in contact. So next what will happen is the enzyme will bind the substrate, but this binding won't be perfect. So I'll call this initial binding, which is stage 2 of the process. And what that means, there's a lot of people that say times the grade equal.
I think I watched all zeros, right?
We create a free MCAT program that's going to have you fully prepared to take your exam in 4 months.
And then we can go home. And then final one.
Is that the forces holding these two together are strong, but they're not at their maximum strength just yet. And enzymes and substrates don't actually fit together quite like puzzle pieces. And they actually work a little bit more like two pieces of clay that will both mold together so that the fit is much tighter. So in our next step, this is exactly what happens. The enzyme and the substrate will both change shape a little bit and bind to each other really strongly. And we call this the induced fit because both the enzyme and the substrate have changed their a little bit more like two pieces of clay that will both mold together so that the fit is much tighter. So in our next step, this is exactly what happens. The enzyme and the substrate will both change shape a little bit and bind to each other really strongly. And we call this the induced fit because both the enzyme and the substrate have changed their shape a little bit so that they bind together really tightly. And it's at this point where the reaction that the enzyme is catalyzing is at full force. And this would be stage 3. So our next stage occurs after the reaction is completed, and the binding becomes similar to what it was in stage 2. But the difference, and we call this the induced together quite like is the enzyme will bind the substrate. But this binding won't be perfect. So I'll call this initial binding, which is stage 2 of the process. And what that means is that the forces holding these two together are strong, but they're not at their maximum strength just yet. And enzymes and substrates don't actually fit together quite like puzzle pieces. And they actually work a little bit more like two pieces of clay that will both mold together so that the fit is much tighter. So in our next step, this is exactly what happens. The enzyme and the substrate will both change shape a little bit and bind to each other really strongly. And we call this the induced fit because both the enzyme and the substrate have changed their shape a little bit so that they bind together really tightly. And it's at this point where the reaction that the enzyme is catalyzing is at full force. And this would be stage 3. So our next stage occurs after the reaction is completed, and the binding becomes similar to what it was in stage 2. But the difference here is that there is something different about the substrate. So in this reaction, the enzyme is cutting our substrate into 2 parts. So now the 2 parts have become separated. And this would occur after the reaction is finished, and we'll call this stage 4. Now, in our next and last stage, the products of the reaction have been released from the enzyme, and our enzyme is back in the same state that it was in stage 1. And we'll call this stage 5. Now let's look at this from a slightly different angle. I'm going to label the enzyme as E, the substrate as S, and our 2 products as P1 and P2. And they're going to represent this series of events as different steps in a sequence of reactions. So first...
Okay, hold on, Mom. Let me blow my nose and get some socks. We're a little behind. We got a lot to do today.
we'll have E and S separate, and this is stage 1. And next, E and S will bind to each other to form an enzyme substrate combo.
What? I'm joking. It was only a joke. Why you gotta get so mad? It's only a game. It's only a game. Why you have to get so mad? Something like that, I think.
Complex, which I've called ES and corresponds to stage 2 from before. Now, what's really interesting is that in the next step, where we had the induced fit of stage 3, we're actually at the transition state of the entire reaction. And this is the same as that really high energy point that we saw at the beginning of this video. And it's at the point of the transition state where our enzyme is most tightly bound to its substrate. Now, I've written the substrate out here with the letter X because at the reaction's transition state, our substrate isn't quite a reactant and it isn't quite our product either. It's somewhere in between. So that's why I've written it out as X instead of S. And I've also written this double dagger symbol, which is just a universal symbol for transition states. Now, in our next stage, which is after the reaction has occurred, since it exists after the transition state, we have the enzyme bound to the two products P1 and P2. And this was stage 4 from before. And then finally, in our last stage, stage 5, we have our enzyme, which is now separated from our two products P1 and P2. Now, the big takeaway from all this is that binding between enzyme and substrate is strongest at the reaction's transition state. And this is because the enzyme and the substrate have molded together, and that's why we call it the induced fit. Now, some enzymes will actually bind more than one substrate. And if we look at a reaction that might be familiar, which is lactic acid fermentation, we can see that our enzyme, lactate dehydrogenase, will have space to bind two different substrates in this reaction. One space being for NADH and the other being for pyruvate. So enzymes don't necessarily bind just one substrate. Now, sometimes things will bind to enzymes at places other than their active sites, and we call this allosteric binding. So if we have an enzyme here with its active site, a regulating molecule, like an inhibitor, may bind the enzyme at a different location than the enzyme's active site. Now, when something binds to an enzyme like this, it usually has the effect of changing the shape of an enzyme in some way to affect its ability to catalyze reactions. So in this case, when an inhibitor binds the enzyme, it might change the shape of the active site, thereby inhibiting the enzyme as it's no longer able to bind its intended substrate, since they don't quite fit together anymore. So while enzymes bind reactive groups at their active sites, they can also bind regulators at their allosteric sites, and allosteric sites just refer to any binding site outside of the active site. And remember, allosterically binding molecules can either be activators or inhibitors, any regulating molecule. So what did we learn? Well, first we learned that enzymes are specific, and that they can each bind to only specific substrates to catalyze specific reactions. Next, we learned about the induced fit model, and how enzymes bind their substrates most tightly in the middle of a reaction at the reaction's transition state. And finally, we learned that enzymes have both active sites and allosteric sites, with active sites being where the reaction takes place and allosteric sites being where regulation takes place. Today, we're going to talk about cofactors and coenzymes, and how sometimes they can be essential to proper enzymatic function. But first, let's review the idea that enzymes make reactions go faster. And they do this by lowering the activation energy peak of their respective reactions. Let's also review the idea that enzymes bind their substrates at a location on the enzyme called the active site, which is where most of the reaction takes place. Now, not all enzymes are able to catalyze reactions on their own, and some need a little help. So if we have our enzyme here trying to react with our substrate over here, sometimes something called a cofactor or a coenzyme will be needed, which will also need to bind to the enzyme in order for it to function properly. And we're going to go over what coenzymes and cofactors are and exactly how they work. So first, we'll talk about what a coenzyme is. Well, coenzymes are organic carrier molecules. And what I mean by organic is that they're primarily carbon-based molecules. And by carrier, I mean that coenzymes hold on to certain things for an enzyme to make the catalysis run a little more smoothly. And a great example of a coenzyme is NADH, which acts as an electron carrier. And here I've shown NADH dissociating into its oxidized form, NAD+, as well as to a hydride ion, which basically just exists as a pair of electrons that some other molecule would be grabbing. So NAD+ can accept electrons, causing the molecule to be converted to NADH, which could then carry electrons for an enzyme. Now, if you remember the lactic acid fermentation reaction, where pyruvate is converted to lactic acid, you'd see that the enzyme catalyzing this reaction, lactate dehydrogenase, uses NADH as a coenzyme in order to transfer electrons to the pyruvate molecule in order to turn it into lactic acid. And in this sense, NADH is acting as an electron-carrying coenzyme. Another example of a coenzyme is coenzyme A, which, like NADH, acts as a carrier molecule, but instead of carrying electrons like NADH does, coenzyme A, which we sometimes call CoA, holds on to acyl or acetyl groups instead. And you'd see CoA appear quite often in metabolic reactions, where it'll carry these two carbon acetyl groups from one molecule to another. Now, cofactors are a little different from coenzymes. While coenzymes are only really involved in transferring different things from one molecule to another, cofactors are directly involved in the enzyme's catalytic mechanism. They don't strictly carry something like a coenzyme would, but might be stabilizing the enzyme or the substrates, or helping the reaction convert substrates from one form to another. A great example of this is with the enzyme DNA polymerase. Remember that DNA polymerase is responsible for helping out with synthesizing new DNA during DNA replication. Now, you may remember that DNA is a very negatively charged molecule because of all the negatively charged phosphate groups that you'll find around it. Well, DNA polymerase uses a magnesium ion as a cofactor, which can use its big positive charge to stabilize all that negative charge on DNA. And you can see how this is different from a coenzyme, because instead of acting as a carrier molecule, the magnesium ion cofactor is stabilizing the DNA and is more directly involved in the actual catalysis. Now, interestingly, what people normally call vitamins and minerals, like the kinds that a doctor would tell you to make sure you get enough of in your diet, are often different cofactors and coenzymes. And what's special about vitamins and minerals is that your body can't build them up from scratch, and you need to get them from your diet in order to stay healthy. So when we say vitamins, we typically refer to organic cofactors and coenzymes. So two great examples are ones we just discussed. Vitamin B3, which you may see being called niacin on a food label, is actually just a precursor for NAD. And vitamin B5 is just a precursor for coenzyme A. Minerals, on the other hand, are inorganic, meaning they aren't carbon-based, and minerals are usually just cofactors in our body. So magnesium would be a great example of a mineral cofactor that an enzyme like DNA polymerase would use. Now, not all minerals act only as cofactors. Some minerals, like calcium, which can act as a cofactor, is also a critically important component of bone and teeth. And it doesn't strictly act as an enzyme cofactor here. It's actually an important part of the structure itself. So what did we learn? Well, first we learned that not all enzymes are able to function alone, and some need a little help. And next we learned that this help can come from coenzymes, which usually act as carrier molecules, or cofactors, which directly assist with the catalysis the enzyme is doing. And finally, we learned that vitamins and minerals generally refer to dietary cofactors and coenzymes. So today we're going to talk about enzymes and all the different kinds of reactions that enzymes can catalyze. But before we do that, let's review the idea that enzymes make biochemical reactions go faster. And if you look at a reaction coordinate diagram, you'd notice that enzymes speed up reactions by lowering their activation energy. Now, enzymes are generally named for their reactions, which is convenient because it makes it a lot easier to remember what an enzyme does if someone gives you its name. And a great example of this is that one of the enzymes involved in DNA replication is called DNA polymerase, which is named as such because it acts on DNA and specifically makes polymers of DNA. Now, the suffix ase is usually just one that you find at the end of most enzyme names. Now, another great example is that the enzyme that catalyzes the first step of glycolysis, which you may remember is the reaction between glucose and ATP to form glucose 6-phosphate and ADP, is called hexokinase. And hexo refers to the number 6, which is a reference to glucose being a 6-carbon sugar, and kinase is a term referring to enzymes that add phosphate functional groups to different substrates. So overall, hexokinase adds phosphates to 6-carbon sugars like glucose. Now, generally, every enzyme has a very specific name that gives insight into the specific reaction that that enzyme can catalyze. So we can actually divide most enzymes into 6 different categories based off the kinds of reactions that they catalyze. Now, our first group is the transferase group. And the basic reaction that transferases catalyze are ones where you move some functional group X from molecule B to molecule
Okay, we gotta spend a little bit of time to actually memorize this. I feel like if we can get to memorize these and then watch the rest of the videos, we're done with the continuity for this one. And then we'll move on to kinetics, which doesn't have a lot. But, could be deceiving. Oh man, it's crazy. Oh wow.
A. And a great example of one of these reactions occurs during protein translation, where amino acids bound to tRNA molecules are transferred over to the growing polypeptide chain. So in this case, A refers to our amino acid chain, B refers to our tRNA, and X refers to this lysine residue, which is being transferred from B to A. And this reaction in particular is catalyzed by an enzyme called peptidyl transferase, which is an appropriate name since it is a transferase involved in making peptides. Next, we have the ligase group, which catalyzes reactions between two molecules, A and B, that are combining to form a complex between the two, or AB. And an example of a reaction using a ligase that you might be familiar with occurs during DNA replication, where two strands of DNA are being joined together. So in this reaction, A and B represent the two separated DNA polymers, which are being joined to form a single strand. And this reaction in particular is catalyzed by an enzyme called DNA ligase, which is named since it's a ligase that works on DNA strands. Now, our third group is the oxidoreductase group, which is a little different from the others since it actually includes two different types of reactions. And these reactions involve transferring electrons from either molecule B to molecule A, or from molecule A to molecule B. Now, we say that an oxidase is directly involved in oxidizing or taking electrons away from a molecule, while a reductase is involved in reducing or giving electrons to a molecule. And we call these enzymes
Hold on, buddy. Man, I'm sneezing like a storm. Let's do 10 minutes on the clock. Anybody ask me anything? No. Good. back. You're back. Put another 60 on the clock. Put another 60 on the clock. What is this? Alrighty. Gotta get to it. I need to wash my hands, I guess. Maybe that's why it's
together because they can.
Whoa, buddy. Apologies right now, man. Right now. Because real quick. Looks like you're whatever. let's just memorize the chart. You need to also memorize what subtypes they belong to. I mean, what's the subtypes that they belong to for which class? Well, this new need to keep it 12. It's early, but whatever. I keep having a runny nose, man. Not really that cool. Let's get some more water. So memorize the 6 enzymes, the subclasses, like some specific enzymes, I guess, that belong to the type of enzyme or the class. And then we'll rewatch the video. And once I'm able to say which ones would be based on that class, we'll move on. And then we'll go ahead and move on to the next section, right? And follow the plan for today. I think it's too hot now. Maybe it's getting too hot. I don't know. Okay, that's a little better. I'm gonna take the next one. It's just too much. All right, that's good. You know what I want? I do that right now. I want to get not look at this later. Yes, yeah. I think. I'm not sure if that's all where I'm at right now. Let's see. This one. Yeah. Protease? 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 Protease. Lipase. Oh shoot. Oh wait, no, that's true. Oh my goodness. Okay. I'm not sure. So we're missing second one for lyase. Yeah. The second one for lyase, hydrolase. Possibly the second one for oxidoreductase. Oh, aldolase and decarboxylate. That is in the lyase. And then protease for hydrolase is protease. And lipase. That's pretty short. Oxidoreductase is there electron transfer. Dehydrogenase, oxidase. Oxidase. Um, close, I think. Mhm. Lyase, lyase. Lyase. Carboxylate. 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 Who? Maybe I'll get it once I start writing it down. I didn't get it. It was missing one. We are missing one. Oh, aldolase. Aldolase. Oh, we were close. We were literally 1.5. Aldolase and carboxylate. Carboxylase. Lace, not eight. Lace, ASE. Good my enzymes here, buddy. Tom's. Yeah, I'm happy to do it. try again. Let's go. How many should I do? I'll do this. I got one, so let's see if I can do the video, be ahead of it, and then after the video, then I'll do it one more time, and then we'll move on.
Talk about enzymes and all the different kinds of reactions that enzymes can catalyze. But before we do that, let's review the idea that enzymes make biochemical reactions go faster. And if you look at a reaction coordinate diagram, you'd notice that enzymes speed up reactions by lowering their activation energy. Now, enzymes are generally named for their reactions, which is convenient because it makes it a lot easier to remember what an enzyme does if someone gives you its name. And a great example of this is that one of the enzymes involved in DNA replication is called DNA polymerase, which is named as such because it acts on DNA and specifically makes polymers of DNA. Now, the suffix ase is usually just one that you find at the end of most enzyme names. Now, another great example is that the enzyme that catalyzes the first step of glycolysis, which you may remember is the reaction between glucose and ATP to form glucose 6-phosphate and ADP, is called hexokinase. And hexo refers to the
Kinase. kinase, it transfers functional groups from one molecule to another. Kinase and aminotransferase.
Number 6, which is a reference to glucose being a 6th carbon sugar, and kinase is a term referring to enzymes that add phosphate functional groups to different substrates.
Oh, kinase add phosphate groups. I didn't know that part particularly. It's not too bad though.
So overall, hexokinase adds phosphates to 6 carbon sugars like glucose. Now, generally, every enzyme has a very specific name that gives insight into the specific reaction that that enzyme can catalyze. So we can actually divide most enzymes into 6 different categories based off the kinds of reactions that they catalyze. Now, our first group is the transferase.
Transfer functional groups to other molecules. Kinase, for example, will transfer against phosphate groups. Will transfer phosphate groups. Adding phosphate groups to the molecule. We have kinase. We also have aminotransferase
group. And the basic reaction that transferases catalyze are ones where you move some functional group X from molecule B to molecule A.
Okay.
And a great example of one of these reactions occurs during protein translation, where amino acids bound to tRNA molecules are transferred over to the growing polypeptide chain. So in this case, A refers to our amino acid chain.
Kinase adds phosphate groups using ATP to substrates.
B refers to our tRNA, and X refers to this lysine residue, which is being transferred from B to A. And this reaction in particular is catalyzed by an enzyme called peptidyltransferase, which is an appropriate name since it is a transferase involved in making peptides. Next, we have the ligase group, which catalyzes
ligase, links or glues, one molecule to the other, using ATP, for example, DNA ligase or synthetase. Synthetase forms new bonds with ATP. DNA ligase.
Reactions between two molecules.
Well, I guess that'd be involved in adding DNA
molecules A and B that are combining to form a complex between the two, or AB. And an example of a reaction using a ligase that you might be familiar with occurs during DNA replication, where two strands of DNA are being joined together. So in this reaction, A and B represent the two separated DNA polymers, which are being joined to form a single strand. And this reaction in particular is catalyzed by an enzyme called DNA ligase, which is named since it's a ligase that works on DNA strands. Now, our third group is the oxidoreductase group, which is a little different from the others.
Oxidoreductase will transfer electrons or undergo redox reactions. We have give me a second. Dehydrogenase. Oh, goodness. Dehydrogenase. And oxidase. Dehydrogenase and oxidase.
Since it actually includes two.
Dehydrogenase transfers electrons using NAD and FAD. NAD plus or FAD. Transfer electrons or protons. Oxidase is not there.
Two different types of reactions. And these reactions involve transferring electrons from either molecule B to molecule A, or from molecule A
molecule A and molecule B.
to molecule B.
Mm-hmm.
Now, we say that an oxidase is directly involved in oxidizing or taking electrons away from a molecule,
Right.
while a reductase is involved in reducing or giving electrons to a molecule.
Right.
And we call these enzymes oxidoreductases together because they can usually catalyze both the forward and reverse reactions, which is why I've used equilibrium arrows here instead of just a normal single-headed arrow. Now, a great example of an oxidation-reduction reaction occurs during lactic acid fermentation, where electrons are either passed from NADH to pyruvate or from lactic acid to NAD. Now, this reaction is catalyzed by an enzyme called lactate dehydrogenase. Remember that the word dehydrogenase refers to the removal of a hydride functional group. And that's the same as saying the removal of electrons, since hydrides are basically just hydrogen atoms with two electrons on them instead of just one. Now, this enzyme is given its name since it's able to remove a hydride, or remove electrons, from a molecule of lactic acid. Next, we have the isomerase group. And enzymes in this group are typically
isomerase. We have aldolase and No. We have phosphoglucose isomerase, which will rearrange the molecules without adding or removing anything.
Involved in reactions where a molecule like molecule A is being. Next, we have the isomerase group. And enzymes in this group are typically involved in reactions where a molecule like molecule A is being converted to one of its isomers. And an example of this type of a reaction is the conversion of glucose 6-phosphate to fructose 6-phosphate, which is one of the steps of glycolysis that you may remember. Now, this reaction is catalyzed by an enzyme called phosphoglucose isomerase, which is appropriately named since it creates isomers of glucose molecules that are phosphorylated. Now, our next category is the hydrolase category. And hydrolases use
hydrolase. So you have hydrolase. We have lipase and protease. Protease and lipase.
Water to cleave a molecule like molecule A into two other molecules, B and C. And a great example of one of these reactions is the hydrolysis reaction that can occur to peptide bonds. And if we have this lysine-alanine dipeptide here, it could be reacted with water to form two individual amino acids that are no longer bound. And this particular hydrolysis reaction can be catalyzed by a class of enzymes that we call serine hydrolases, which some people call serine proteases. And they are named this way because they are hydrolases that use a serine residue as the key catalytic amino acid that is responsible for breaking the peptide.
Oh man. Hydrolase. Lipase. typos. Protease or hydrolase. What the heck? Well, protease is also hydrolase. It's kind of in the name, huh?
Bond. Now, our last category is a little more complicated than the others.
Okay,
And it's
lyase breaks bonds without water or redox. We have aldolase and carboxylase.
the lyase group. Now, lyases catalyze the dissociation of a molecule like molecule A into molecule B and C without using water like hydrolases would, and without using oxidation or reduction like an oxidoreductase would. And one example of a reaction catalyzed by a lyase is the cleavage of argininosuccinate into arginine and succinate.
Fumarate.
And this reaction takes place
Arginine
during
and
the
fumarate.
urea cycle, which you also might be familiar with. Now, this specific reaction is catalyzed by an enzyme called argininosuccinate lyase,
Didn't take our
which is
dance.
appropriately named because it is a lyase that catalyzes the breakdown of an argininosuccinate molecule. Now, it's important to recognize that since lyases don't use water or oxidation to break a bond, they need to generate either a double bond between two atoms or a ring structure in a molecule in order to work. So, what did we learn? Well, first we learned that enzymes are sometimes named for their reactions. And next we learned about the 6 different types of enzymes. We have transferases, which transfer functional groups from one molecule to another, ligases, which ligate or join two molecules together, oxidoreductases, which move electrons between molecules, isomerases, which convert a molecule from one isomer to another, hydrolases, which break bonds using water, and lyases, which break bonds without using water and without using oxidation.
Oh shoot, it was decarboxylase, not carboxylase. Nah, we have to go again. Almost. I feel like I'm 80% with this. We gotta get to 100. It's 100 nothing. Ah. Wake up! Wake up! Wake up! We gotta tighten this, dude. We're almost there. We're almost there, but we gotta kind of hurry up. All right, let's go again. Ligase. We're gonna link or glue molecules together using ATP. Here we got DNA ligase or synthetase, which will synthetase form something. I'm not sure. Form synthesis. Form blank using ATP. Isomerase. We're going to rearrange molecules without rearrange molecules without rearrange molecules without adding or removing anything. Not adding or removing molecules, I guess, right? Isomerase, we have phosphoglucose isomerase, which is some sort of isomer, right? We're creating an isomer. Example is an isomer. Lyase. We are breaking bonds without water or redox. Without water or redox. Lines we have aldolase and decarb. Yeah, and decarboxylase. Hydrolase. Breaking bonds with water. Breaking bonds with water. We have example is serine hydrolase, or protease, and lipase. Okay, oxidoreductase is transferring electrons or protons between different molecules. We have oxidase and hydrogen. Not hydrogen. Wait a minute. What's the name? Hydrogenase? Is it hydrogenase? I'm not sure. Hydrogenase transfers functional groups between molecules. Transferase, we have aminotransferase and and one more. Seriously? Oh, and kinase. Kinase will add the functional group phosphates. Well, I guess what? Oh yeah, phosphates to the molecule. Okay. Hydrogenase. Hydrogenase was for what he said? I'm not sure, but so hydrogenase, and review what it was for. I think he gave another example. The oxidoreductase, hydrogenase, and retransferase. Transferase subtypes. That's not going down. Okay, now I think we're like 90%.
So today we're going to talk about enzymes and all the different kinds of reactions that enzymes can catalyze. But before we do that, let's review the idea that enzymes make biochemical reactions go faster. And if you look at a reaction coordinate diagram, you notice that enzymes speed up reactions by lowering their activation energy. Now, enzymes are generally named for their reactions, which is convenient because it makes it a lot easier to remember what an enzyme does if someone gives you its name, most enzyme names. Now, another great example is that the enzyme that catalyzes the first step of glycolysis, which you may remember is the reaction between glucose and ATP to form glucose 6-phosphate and ADP, is called hexokinase.
Oh, hexokinase. Okay.
And hexo refers to the number 6, which is a reference to glucose being a 6th carbon sugar, and kinase is a term referring to enzymes that add phosphate functional groups to different substrates. So overall, hexokinase adds phosphates to 6 carbon sugars like glucose. Now, generally, every enzyme has a very specific name that gives insight into the specific reaction that that enzyme can catalyze. So we can actually divide most enzymes into 6 different categories based off the kinds of reactions that they catalyze. Now, our first group is the transferase group. And the basic reaction that transferases catalyze are
Transferase, transfer functional groups of different molecules. We have kinase was the one he just mentioned, involved in glycolysis, and we have aminotransferase.
are ones where you move.
An example would be that he's going to give is the peptidyl transferase.
Some functional group X from molecule B to molecule A. And a great example of one of these reactions occurs during protein translation, where amino acids bound to tRNA molecules are transferred over to the growing polypeptide chain. So in this case, A refers to our amino acid chain, B refers to our tRNA, and X refers to this lysine residue, which is being transferred from B to A. And this reaction in particular is catalyzed by an enzyme called peptidyl transferase, which is an appropriate name since it is a transferase involved in making peptides. Next, we have the ligase group, which catalyzes reactions between two molecules.
Ligase, ligase is going to ligase will link or glue molecules together using ATP. DNA ligase, synthetase.
A and B that are combining to form a complex between the two, or AB. And an example of a reaction using a ligase
Synthetics will form bonds with ATP.
that you might be familiar with occurs during DNA replication, where two strands of DNA are being joined together. So in this reaction, A and B represent the two separated DNA polymers, which are being joined to form a single strand. And this reaction in particular is catalyzed by an enzyme called DNA ligase, which is named since it's a ligase that works on DNA strands. Now, our third group is the oxidoreductase.
Oxidase. Oxidase and hydrogenase. Oxidoreductase will transfer electrons and protons between the different molecules.
Base group, which is a little different from the others since it actually includes two different types of reactions. And these reactions involve...
It is the hydrogenase. Okay, we're good. It is the hydrogenase. Is that what I said? No, I said hydrogenase. It needs to be dehydrogenase. Dehydrogenase.
While transferring electrons from either molecule B to molecule A, or from molecule A to molecule B. Now, we say that an oxidase is directly involved in oxidizing or taking electrons away from a molecule, while a reductase is involved in reducing or giving electrons.
Oh, Rig, oxidation is loss of electrons. Rig, reduction is gain of electrons. Hey,
To a molecule.
so
And we call these enzymes oxidoreductases together because they can usually catalyze both the forward and reverse reactions, which is why I've used equilibrium arrows here instead of just a normal single-headed arrow. Now, a great example of an oxidation-reduction reaction occurs during lactic acid fermentation, where electrons are either passed from NADH to pyruvate or from lactic acid to NAD. Now, this reaction is catalyzed by an enzyme called lactate dehydrogenase. Remember that the word dehydrogenase refers to the removal of a hydride functional group. And that's the same as saying the
removal of a hydride functional group. Removal
of electrons.
Removal of an electron. Oxidation.
Hydrides are basically just hydrogen atoms with two electrons on them instead of just one.
Gotcha.
Now, this enzyme is given its name since it's able to remove a hydride or remove electrons from a molecule of lactic acid. Next, we have the isomerase group. Now, this enzyme is given its name since it's able to remove a hydride or remove electrons from a molecule of lactic acid. Next, we have the isomerase group.
Isomerase, phosphoglucose isomerase. Will rearrange molecules. Without adding or removing anything.
And enzymes in this group are typically involved in reactions where a molecule, like molecule A, is being converted to one of its isomers. And an example of this type of a reaction is the conversion of glucose 6-phosphate to fructose 6-phosphate, which is one of the steps of glycolysis that you may remember. Now, this reaction is catalyzed by an enzyme called phosphoglucose isomerase, which is appropriately named since it creates isomers of glucose molecules that are phosphorylated. Now, our next category is the hydrolase category. And
hydrolase, we have hydrolase. We have aldolase and lipase. Aldolase and lipase.
Hydrolase is used.
No. We have hydrolase, protease, and lipase. Hydrolase, protease, lipase, protease.
Water to cleave a molecule like molecule A into two other molecules, B and C. And a great example of one of these reactions is the hydrolysis reaction that can occur to peptide bonds, And if we have this lysine-alanine dipeptide here, it could be reacted with water to form two individual amino acids that are no longer bound. And this particular hydrolysis reaction can be catalyzed by a class of enzymes that we call serine hydrolases, which some people call serine proteases. And they are named this way because they are hydrolases that use a serine residue as the key catalytic amino acid that is responsible for breaking the peptide bond. Now, our last category is a little more complicated than the others, and it's the
Okay, lyase. Aldolase and decarboxylase.
lyase group. Now-
Lyase group will break bonds without water and without redox and without redox reaction.
Lyases catalyze the dissociation of a molecule, like molecule A, into molecule B and C without using water, like hydrolases would, and without using oxidation or reduction, like an oxidoreductase would.
Oxidation reduction.
And one example of a reaction catalyzed by a lyase is the cleavage of argininosuccinate into arginine and succinate.
Fumarate.
And this reaction takes place during the urea cycle, which you also might be familiar with. Now, this specific reaction is catalyzed by an enzyme called argininosuccinate lyase, which is appropriately named because it is a lyase that catalyzes the breakdown of an argininosuccinate molecule. Now, it's important to recognize that since lyases don't use water or oxidation to break a bond, they need to generate either a double bond between two atoms or a ring structure in a molecule in order to work. So, what did we learn? Well, first we learned that enzymes are substances which transfer oxidoreductases, which move electrons between molecules, and lyases, which break bonds without using water.
All right, let's take a 10-minute. We'll hit it again. Hopefully I get it all right. And then we can move on. You said nice stuff, man. Thanks, Rabia. Oops, God. back. All right, we'll run through it one more time. Hopefully I get it this time. Watch this video, and then we just gotta move on. We just gotta move on. Let's do the black one for now. I think that was a bit too much. Get him, cops, get him. Get him, cops, get him. All right, we got line. Oh. All right. We got ligase. That's gonna link or glue molecules together using ATP. Examples are DNA ligase or synthetase. DNA ligase. Okay. We have isomerase. We have rearrangement of molecules, rearrangement of atoms, without adding or removing anything. Examples are phosphoglucose isomerase, lyase, lyase. Is that what it's called? It's called lyase? Hold on. Lyase? That's it? Just lyase? Oh no, just lyase. Okay. Lyase, we have breaking bonds without water or oxidation or reduction, or redox. Examples for this one are aldolase and decarboxylase. Hydrolase. We have breaking bonds with water. Example is serine hydrolase or protease. And protease and lipase. Oxidoreductase. We have transferring electrons or protons between molecules. Examples are dehydrogenase and oxidase. Transferase. We have transferring functional groups between molecules. Transferring functional groups between molecules. Examples just have amino transferase or kinase, and kinase will add phosphate groups to the molecule. I think so. That's what I got. Let's check it.
Reactions that enzymes can catalyze. But before we do that, let's review the idea that enzymes make biochemical reactions go faster. And if you look at a reaction coordinate diagram, you'd notice that enzymes speed up reactions by lowering their activation energy. Now, enzymes are generally named for their reactions, which is convenient because it makes it a lot easier to remember what an enzyme does if someone gives you its name. And a great example of this is that one of the enzymes involved in DNA replication is called DNA polymerase.
Right.
Which is named as such because it acts on DNA and specifically makes polymers of DNA. Now, the suffix ase is usually just one that you find at the end of most enzyme names. Now, another great example is that the enzyme that catalyzes the first step of glycolysis, which you may remember is the reaction between glucose and ATP to form glucose 6-phosphate and ADP, is called hexokinase. And hexo refers to the number 6, which is a reference to glucose being a 6-carbon sugar, and kinase is a term referring to enzymes that add phosphate functional groups to different substrates. So overall, hexokinase adds phosphates to 6-carbon sugars.
Oh. That's a transferase. Hexokinase is a transferase because it's kinase. So there's hexokinase.
Just like glucose. Now, generally, every enzyme has a very specific name. To glucose being a 6-DP, it's called hexokinase, is that the enzyme that catalyzes DNA and specifically makes polymers of DNA. Now, the suffix ase is usually just one that you find at the end of most enzyme names. Now, another great example is that the enzyme that catalyzes the first step of glycolysis, which you may remember is the reaction between glucose and ATP to form glucose 6-phosphate and ADP, is called hexokinase. And hexo refers to the number 6.
Hexokinase, first step of glycolysis.
Which is a reference to glucose being a 6-carbon sugar, and kinase is a term referring to enzymes that add phosphate functional groups to different substrates. So overall, hexokinase adds phosphates to 6-carbon sugars like glucose. Now, generally, every enzyme has a very specific name that gives insight into the specific reaction that enzyme can catalyze. So we can actually divide most enzymes into 6 different categories based off the kinds of reactions that they catalyze. Now, our first group is the transferase group, and the basic reaction that transferases catalyze are ones where you move some functional group X from molecule B to molecule A. And a great example of one of these reactions occurs during protein translation, where amino acids bound to tRNA molecules are transferred over to the growing polypeptide chain. So in this case, A refers to our amino acid chain, B refers to our tRNA, and X refers to this lysine residue, which is being transferred from B to A. And this reaction in particular is catalyzed by an enzyme called peptidyltransferase, which is an appropriate name since it is a transferase involved in making peptides. Next, we have the ligase group, which catalyzes reactions between two molecules, A and B, that are combining to form a complex between the two, or AB. And an example of a reaction using a ligase that you might be familiar with occurs during DNA replication, where two strands of DNA are being joined together. So in this reaction, A and B represent the two separated DNA polymers, which are being joined to form a single strand. And this reaction in particular is catalyzed by an enzyme called DNA ligase, which is named since it's a ligase that works on DNA strands. Now, our third group is the oxidoreductase group, which is a little different from the others since it actually includes and this reaction in particular is catalyzed by a little different from the others since it actually includes 2 different types of reactions. And these reactions involve transferring electrons from either molecule E to molecule A, or from molecule A to molecule B. Now, we say that an oxidase is directly involved in oxidizing or taking electrons away from a molecule, while a reductase is involved in reducing or giving electrons to a molecule. And we call these enzymes oxidoreductases together because they can usually catalyze both forward and reverse reactions, which is why I've used equilibrium arrows here instead of just a normal single-headed arrow.
Okay.
Now, a great example of an oxidation reduction reaction occurs during lactic acid fermentation, where electrons are either passed from NAD.
Lactic acid fermentation.
H to pyruvate, or from lactic acid to NAD. Now, this reaction is catalyzed by an enzyme called lactate dehydrogenase. Remember that the word dehydrogenase refers to the removal of a hydride functional group, and that's the same as saying the removal of electrons, since hydrides are basically just hydrogen atoms with 2 electrons on them instead of just 1. Now, this enzyme is given its name since it's able to remove a hydride or remove electrons from a molecule of lactic acid. Next, we have the isomerase group, and enzymes in this group are typically involved in reactions where a molecule like molecule A is being converted to one of its isomers. And an example of this type of reaction is the conversion of glucose 6-phosphate to fructose 6-phosphate, which is one of the steps of glycolysis that you may remember. Now, this reaction is catalyzed by an enzyme called phosphoglucose isomerase, which is appropriately named since it creates isomers of glucose molecules that are phosphorylated. Now, our next category is the hydrolase category, and hydrolases use water to cleave a molecule like molecule A into 2 other molecules, B and C. And a great example of one of these reactions is the hydrolysis reaction that can occur to peptide bonds. And if we have this lysine-alanine dipeptide here, it could be reacted with water to form 2 individual amino acids that are no longer bound. And this particular hydrolysis reaction can be catalyzed by a class of enzymes that we call serine hydrolases, which some people call serine proteases. And they're named this way because they are hydrolases that use a serine residue as a key catalytic amino acid that is responsible for breaking the peptide bond. Now, our last category is a little more complicated than the others, and it's a lyase group. Now, a Lyases catalyze the dissociation of a molecule A into molecule B and C without using water like hydrolases would, and without using oxidation or reduction like an oxidative reductase would. And one example of a reaction catalyzed by a lyase is the cleavage of argininosuccinate into arginine and succinate. And this reaction takes place during the urea cycle, which you also might be familiar with. Now, this specific reaction is catalyzed by an enzyme called argininosuccinate lyase, which is appropriately named because it is a lyase that catalyzes the breakdown of an argininosuccinate molecule. Now, it's important to recognize that since lyases don't use water or oxidation to break a bond, they need to generate either a double bond between two atoms or a ring structure in them.
I didn't know that. I didn't remember that. Generate a double bond or a ring. Lysis generate a double bond.
Molecule in order to work. So what did we learn? Well, first we learned that enzymes are sometimes named for their reactions.
I think that's okay. I mean, it's a little bit of detail. Let me see if I can answer these questions now, I guess. So one, I'm gonna go with transferase. 2, removes a phosphate group. Removes a phosphate group. Removes a phosphate group. From a protein, I'm going to go with hydrolase. Removes a phosphate group. Is it going to be more than one, or is it just, oh, it says one. Removes hydrogens. 3 oxidoreductase. Oh, just what class? Removes a carbon dioxide molecule. Lyase. Shifting the phosphate group's position. Isomerase. You mentioned the urea one, but it says using ATP. I'm gonna go with lyase. Without using ATP? Without using ATP? Lyase. Let's see what I get. Oh, let's go. We actually got them all. Ready? Share screen. This is what I'm using to ask questions. I mean, I don't have an MCAT tutor, but this works pretty good. So it's just create your own project. I think you get plus for 2 months free if you're a student. There's like a promo sale going on or something. So I definitely say to get it for sure. High transferase kinase. ATP to glucose. Well, I said transfer, so that's pretty easy, right? Transferase is kinase, and aminotransferase. Hydrolase removes phosphate from protein, phosphatases, oxidoreductase, dehydrogenase, lyase, decarboxylase, isomerase, mutase, joins ammonia, ligase, synthetase, and forms citrate without ATP, lyase, synthase. Ooh, synthase. That's a good one. Sure. All right, I think we're okay now. I think, I never get 100%, but I think it's a lot better than it was before. So we will move on now. Finally, eh?
And different enzymes will often function ideally in different
Whoa, what happened? Oh, yeah.
environments. So today we're going to talk about the effects of the environment on enzymes and how a changing environment can affect an enzyme's ability to catalyze a reaction. But first, let's review the idea that enzymes make reactions go faster. And looking at a reaction coordinate diagram, you notice that enzymes speed things up by lowering a reaction's activation energy. Now, it's important to recognize that enzymes work best in specific environments. And when I say environment, I can be really referring to many different aspects of an enzyme's surroundings, but right now we're really only going to be focusing on pH and temperature values. So let's take another look at this by imagining that we have this person over here, and he's hungry, so he's eating some food. Now, there are a bunch of different digestive enzymes in this guy's body that are going to help him break down all the food he's eating into tiny usable parts. So first, the food will be in his mouth, and one of the enzymes found inside a human's mouth is called alpha amylase, which is responsible for breaking down complex carbohydrates like starches into small, simple carbohydrates like individual sugars. And alpha amylase is able to work well in your mouth since it functions best at a pH of around 7, which is about the same pH as a human's mouth. Now, moving along, the food that our guy ate is going to go all the way down to his stomach, where a whole different group of enzymes will start breaking down food. Now, one enzyme humans have in their stomachs is called pepsin, which breaks down big proteins into smaller peptides. Now, pepsin will be most active at a pH of around 2, which is also the pH of your stomach, which is so low because of all the stomach acid that you find there. Now, in terms of temperature, both of these enzymes typically work at a temperature of around 37 degrees, which is the same as body temperature. But you can see that these two different enzymes are functioning at different environmental conditions. So what would happen if we took an enzyme and moved it into a different environment? Well, let's first look at the effects of changing the pH of an enzyme's environment and jump right in with an example. So remember that DNA is a very negatively charged molecule because of all the negatively charged phosphate groups that you find on DNA. And in order for the enzyme DNA polymerase to help out with DNA replication, it binds a magnesium ion cofactor, which it uses to stabilize all the negative charge on DNA. Now, under normal pH conditions, the DNA polymerase will hold on to that magnesium ion through an electrostatic interaction between magnesium and one of its aspartate residues, which would be deprotonated and thus negatively charged at neutral pH values. Now, if we were to take DNA polymerase and put it into an environment with a reduced, now under normal pH conditions, the DNA polymerase will hold on to that magnesium ion through an electrostatic interaction between magnesium and one of its aspartate residues, which would be deprotonated and thus negatively charged at neutral pH values. Now, if we were to take DNA polymerase and put it into an environment with a reduced pH, then that aspartate residue will become
That's negatively charged. And what do you say?
and put it into an environment with residues, Na. Now, under normal pH conditions, the DNA polymerase will hold on to that magnesium ion through an electrostatic interaction between the magnesium and one of its aspartate residues,
Okay.
which would be deprotonated.
Yeah, at low pH. At low pH. At low pH, H plus 6 like glue. At high pH, H plus will say goodbye.
And thus negatively charged at neutral pH values. Now, if we were to take DNA polymerase and put it into an environment with a reduced pH, then that aspartate residue will become protonated.
The pKa value of aspartic acid is 4. pH value, he said at neutral pH is 7. pH is 7. Aspartic acid is 4. pH is high. H plus will say goodbye. So it'll be what is it?
Action between the magnesium and one of its aspartate residues, which would be deprotonated and thus.
Yeah, BD partner, that's negative.
Must negatively charge at neutral pH
That's
values.
correct.
Now, if we were to take DNA polymerase and put it into an environment with a reduced pH, then that aspartate residues.
It's gonna be proteinated.
Now, if we were to take DNA polymerase and put it into an environment with a reduced pH, then that aspartate residue will become protonated since the pH has dropped so much. And in its protonated form, aspartate no longer has a negative charge and can't hold on to that magnesium ion anymore.
It's gotta go.
And overall, this means that DNA polymerase won't be able to do its job properly in a low pH environment.
DNA polymerase will not be able to do its job properly in a reduced pH environment. Dang, this guy's spitting.
And keeping
Facts. Straight
this enzyme
facts.
at an appropriate pH is essential to its normal function. So now let's take a look at the effects of magnesium ion anymore. And overall, this means that DNA polymerase won't be able to do its job properly in a low pH environment.
Yes, sir.
And keeping this enzyme at an appropriate pH is essential to its normal function.
Okay.
So now let's take a look at the effects of temperature changes on enzyme function. So remember that proteins need to fold into their secondary, tertiary, and possibly quaternary structures in order to be in their functional form.
Right.
And significant changes to a protein's temperature can disrupt a protein's folded geometry and cause it to lose its functionality. If we have our same person from before who's really hungry and really wants to eat, but now this person gets sick with a fever, his temperature will rise and a bunch of the digestive enzymes in his body will get all jumbled up and won't be properly folded anymore. And this is why you might have a hard time eating and digesting food when you have a fever. And this can sometimes lead you to throwing up anything you'll eat, since all of the digestive enzymes in your body won't work anymore because of the increase in your body's temperature. And the food you eat will just sit there in your body and make you feel sick. So what did we learn? Well, first we learned that enzymes generally function only under very specific environmental conditions. And different enzymes will often function ideally in different environments from other enzymes. And next we learned that changes to an enzyme's environment, like changes to the surrounding pH or temperature, can lead to a loss of enzyme functionality.
Let's go ahead and make sure that I added these all correctly to the decks. Because we will review these again today, and then every day, right? But let's make sure that I properly added them before we kind of like really move on. I'm not trying to solve anything. I know we have to move on, but I'm just saying. Oh, really? Did it synchronize? Are you sure? Are you sure it's not much? Really? I don't think so, buddy. I guess it is that many cards. Sure. Brown, we'll type in oxido reductase. Preview. Yeah. That one was added, transferase. Pretty sure that one was added as well. Hydrolase. Yeah, that was added. Liases was needed as well. Isomers. Isomers. Ooh, was this one in? I don't know. That's a YouTube link.
Is convert.
I'll watch the video right now. I'm not gonna watch the video, but I will add this. I will add this. You know, maybe I'll add it once I actually get the glycolysis. And I'll actually watch the video, but for now, it's fine. Isomerase, ligases. Oh, ligase. I don't think so. But let's watch the video and see where it goes. Okay. I don't think we're here yet. I'm not gonna do that one right now. No, thank you. Alright, that's all of them. You know, I'm just gonna add all these. Yeah. All right, there we go. Let's synchronize. Okay, so we add a little bit more cards. That's fine. That's fine. Okay, good. Good, good, good. Good, good, good.
ahorita el pancake, papá?
Yeah.
So in primary ciliary dyskinesia, you can see there's some sort of dyskinesia or problem in movement for the cilia. And mutations in a particular dynein protein lead to this rare disease in which the action of the cilia of the cells lining the respiratory tract fail to function. And this leads to a decrease in mucus clearance from the lungs.
Yay, we're finally on enzyme kinetics. All right. Matthew McConaughey. All right. Let's eat the pancake first on the bacon. And the bacon. And then we'll, move on from there. Watch the video.
So we're going to talk about enzyme kinetics today.
These guys always look too
But
fast.
first, let's review the idea that enzymes speed up reactions by lowering the delta G of the transition state, or lowering the activation energy of a reaction. And also remember that for this to happen, the reacting substrate, which I've called S, will bind to the enzyme E to form the enzyme substrate complex ES before being turned into product P. Also remember that enzymes aren't used up when they catalyze reactions, and that's why we have this E at the end of the equation. Now, there's a general method of thinking that we want to use when talking about enzyme kinetics, and that's what I'm going to talk about today. Now, when we think about kinetics, we want to simplify a reaction as a change from A to B. And you may remember that the rate of this change from A to B would be equal to some rate constant K, which is dependent on the A. About kinetics, we want to simplify a reaction as a change from A to B. And you may remember that the rate of this change from A to B would be equal to some rate constant K which is dependent on the environment of the reaction, multiplied by the concentration of our starting material A. So for our sequence that I mentioned before, E plus S going to ES going to E plus P, we have two reactions going. Going on 1 and 2, which would each have their own rate equation. Rate 1 would be equal to rate constant K1 times the two starting material. Material concentrations, which are E and S, while rate 2 would be equal to K2 times the
Oh, I think that was just it, right? DNA and DNA replication. There's no just DNA here. So I'm guessing it's that one. Okay.
1 would be equal to rate constant K1 times the two starting material concentrations, which are E and S, while rate 2 would be equal to K2 times the concentration of one starting material, ES. Notice that the reaction 1 has two reactants, E and S, while reaction 2 only has one, which is the ES complex. So what do we mean when we say rate? Well, the rate of a reaction is the speed that the reaction goes at, and we can also call it V, which is the symbol for speed. And for our entire reaction of transitioning S to P, our product, the speed would be equal to the rate of change of our concentration of product with respect to time. Or for those of you who aren't really big fans of calculus, the change in P, delta P, over change in time delta T. So to increase the rate that we get new product, we could do this by either increasing the substrate concentration concentration or by increasing the enzyme concentration. Since we're going to assume that the K value is constant and can't be changed. Now, when we think about enzyme kinetics, constant and can't be changed. Now, when we think about enzyme kinetics, we like to assume that we're in a situation where the total concentration of enzyme is constant. And this is generally the case when we're looking at enzymes working in different cells. Now, if we say that we only have 4 enzymes here, and each enzyme can work at a speed of about 10 reactions per second, that would mean that the absolute maximum rate of our reaction would be 40 reactions per second. And this rate we would call Vmax, or
That's right.
max speed.
And speed.
The idea here is that at really high concentrations of substrate, the enzymes will be saturated and full up with substrate and won't be able to react any more quickly. And even if we were to really increase the concentrations of substrate a lot, there will still be a Vmax. there's only so much that we can increase the rate of reaction by increasing the substrate concentration. If we were to look at a graph and plotted the reaction rate V versus our concentration of substrate, we would see that as our substrate concentration got really, really high, the rate would level off as it approached our Vmax value. So when we think about enzymes and their kinetics this way, we've made a couple of assumptions about how our enzymes and substrates are behaving. And I want to talk about these for a moment. The first assumption we've made is that our solutions are behaving ideally.
Okay.
And that we can actually classify our enzyme's reaction into two distinct steps. The first being the binding of substrate to enzymes, and the second being the transitions from substrate to product with the enzyme's help. And by assuming that our solutions are behaving ideally and that we don't have any external factors messing things up, we can simplify our discussion of kinetics quite a bit. Our second assumption is that our two big constants stay constant. We're assuming that our enzyme concentration isn't changing from things like protein synthesis and degradation. And we're also assuming that our rate constant K isn't changing from environmental factors like changes in temperature. Our final assumption is that for our reaction, substrate isn't forming product without the help of enzyme at a big enough rate for us to consider. Our final assumption is that for our reaction, substrate isn't forming product without the help of enzyme at a big enough rate for us to consider. And that
Oh. Gotcha.
it's negligible. Remember that enzymes only speed up reactions. So while it's possible for the substrate to form product without an enzyme, we're going to assume that it's not really happening when we're talking about enzyme kinetics. So what did we learn? Well, first we learned that we can classify enzyme catalysis into two important steps. The first is that the enzymes bind the substrate, and then second, the formation of product. And we talked about how each of these steps has a distinct rate. Second, we learned that if we keep the enzyme concentration constant, then there will be a maximum speed Vmax for that reaction.
Okay. Okay. Michael Menton. Jesus. Michaelis Menton.
So today we're going to talk about Michaelis-Menten
Michaelis.
kinetics in a steady state. But first, let's review the idea that enzymes make reactions go faster, and that we can divide the enzyme's catalysis into two steps. First, the binding of enzyme to substrate, and second, the formation of products. And each of these reactions has its own rate. Let's also review the idea that if we keep the concentration of enzyme constant, then a really high substrate concentration Let's also review the idea that if we keep the concentration of enzyme constant, Then at really high substrate concentrations, we'll hit the maximum speed for a reaction, which we call Vmax. So first, we'll talk about the steady state assumption and max. So first, we'll talk about the steady state assumption and what that means. So like I said before, there are two steps to an enzyme's catalysis. Now, when we use the term steady state, what we mean is that we're at a point where the concentration of ES, or enzyme substrate complex, is constant, which means that the formation of ES is equal to the loss or dissociation of ES. Now, notice that I've used equilibrium arrows between these steps, and that was to show the idea that these reactions, like any reaction, can go forwards or backwards. Our enzyme substrate complex doesn't have to form products. It could just as easily dissociate back to an enzyme and a substrate molecule. So I'll call these reverse reactions.
Oh, that's true.
-1 and -2. If we look at that in terms of our rates, we can say that the rate of formation of ES would be the sum of rate 1 and rate -2. Since both of these reactions lead to ES. And
That's true.
and the rate of loss of ES is equal to the sum of rates -1 and 2.
Okay.
Since both of these lead away from ES. Now also remember that products very rarely go back to reactants, since these reactions are usually thermodynamically stable. So rate -2 is going to be so small in comparison to rate 1 that we can also remember that products very rarely go back to reactants, since these reactions are usually thermodynamically stable. So rate -2
Okay.
is going to be so small in comparison to rate 1 that we can really just cross it out. Which means that we can swap out that second double-headed arrow for a single-headed arrow.
Gotcha.
So using this information, let's do some math. Now I'm going to be deriving a new equation. This can get a bit confusing, so don't worry if you have a little trouble with this. Just rewind the video and try watching it a couple more times if you need to. So I'll start out by drawing the same sequence I did before with the three.
Okay.
Three different reactions. And I'll also write out that steady state equation I mentioned before, where we have rates forming ES equal to rates taking away ES.
Yeah. Okay.
Now the first thing I'll do is swap out those rate values for their rate constants times the reactants for those reactions.
Okay. But I didn't. Oh, because rate constant, the rate 2, the rate -1 goes from the induced fit back to enzyme and substrate. So -1 is going to be, you're starting at the induced fit. Okay. Plus K2 E plus P. And same thing with this one.
So rate 1 will be equal to K1 times E times S, and so on for the other two. Next, I'll introduce a new idea and say that the total amount of enzyme available, which we'll call ET or E total, is equal to the free enzyme E plus the enzyme bound to substrate. Yes. And using this equation, I'm going to rewrite the E on the left side of our equation as the total E minus the ES. Which would be equal to the E we had there before. On the right side of the equation, I've just factored out the common term ES.
Gotcha.
Next, I'm just going to expand the left side of the equation. So take a moment to look at that. And now what I'm going to do is I'm going to divide both sides of the equation by K1. So K1 will disappear on our left side, and on our right side, I've put K1 in with all the other rate constants. Now, since all these rate constants are constant values, I'm going to combine them in this expression of k minus 1 plus k2 over k1 into a new term, km, which I'm going to talk a little bit more about later. So in this next line, I've done two things. First, I've thrown in that km value that I just mentioned, but I've also added es times s to both sides of the
ES, S.
equation. Speed of our whole process, which I'll call VO, is equal to the rate of formation of our things, first side, and on our right is our constant m, just mentioned, but I've also added es times s to both sides of the equations, and thus moved it from the left side to the right. In the next line, I've done two things. First, I switched the left sides and right sides of the equation, just to keep things clear. But I've also factored out the common term es on our new left side. And then what I'm going to do is I'm going to divide both sides of the equation by km plus s. So I can move that term to the right side. So So I'll make some more room over here, and now what I'm going to do is remind you that the speed of our whole process, which I'll call VO, is equal to the rate of formation of our product, which we called rate 2 before, which is also equal to K2 times ES. So now using our equation over here, I'm going to multiply both sides of the equation by K2. Now here's where it gets really tricky. Remember that if we're at our max speed, so our reaction speed V, VO is equal to Vmax, which happens when our substrate concentration is really high. Remember that if we're at our max speed, so our reaction speed VO is equal to Vmax, which happens when our substrate concentration is really high, then our total enzyme concentration is going to be equal to EF.
The total enzyme concentration is going to be equal to
S, since all of our enzyme is saturated by substrate,
right?
and there won't be any free enzyme left. So
Right.
K2 times ET instead of times ES would be equal to Vmax.
I should switch the. I should switch the camera.
Instead of being equal to VO, like you see at the top. So I'll make some room here and then sub in K2.
Well, what the heck? What did I do here? Huh? What the? I can't cap it. Oh man, that's funny. That is funny. Okay.
ES for VO and K2E total for Vmax. And then we finally get to our end equation, which is called the Michaelis-Menten equation, and is super important when we talk about enzyme kinetics.
Okay.
So let's take a few steps back and talk about the Michaelis constant. So first, I'll write out the Michaelis-Menten equation, and if you remember, we created this new term which I called Km, but we never really talked about what it meant. So let's get
What is Km, buddy?
get to that. Now, if you bear with me for a moment and pretend that Km is equal to our substrate concentration,
Okay.
then we can sub in that value into our
Yep, it was. So what happened?
Michaelis-Menten equation, which would put 2s on the bottom, the sum of s plus s, and then the s will cancel out and we'll be left with Vmax over 2.
Oh, okay, yeah.
And what this means is that Km, which we call the Michaelis constant, is defined as the concentration of
1/2. Yeah. Km is where the substrate concentration is equal to VO, which is half of the V max. Substrate concentration where VO is equal to half V max.
of substrate at which our reaction speed is half of the Vmax. But we never wrote to our Michaelis-Menten bottom. So Km left Km, which we call the Michaelis constant, is defined as the concentration of substrate at which our reaction speed is half of the Vmax. Then we can sub in that value into our Michaelis-Menten equation, which would put 2s on the bottom, the sum of s plus s, and then the s will cancel out and we'll be left with Vmax over 2. And what this means is that Km, which we call the Michaelis constant,
Okay.
is defined as the concentration of substrate at which our reaction
speed is half of the V max. Oh. Km, the K is constant, is the concentration of substrate at which the reaction speed is half of the Vmax. Is the concentration of substrate where we're at half of the Vmax. So we're here, half the Vmax is we are here. This is Km. Substrates. To VO. VO equal to so one half V max is equal to VO, and that's equal to the Km.
Speed is half of the Vmax. So when VO is equal to 1/2 of Vmax. And if we look at that on a graph from before, you'd see that Km is a substrate concentration specific to our circumstances.
Rate VO is going to be here, which is Vmax over 2.
Where our VO is equal to 1/2 of Vmax. And if we look at that on a graph from before, you'd see that Km is a substrate concentration specific to our circumstances.
So that's the Km right here.
Where our rate is at half of its max. And the lower our Km, the better our enzyme is at working when substrate
Hold on, buddy.
specific to our circumstances. Where our rate is at half of its max. And the lower our Km, the better our enzyme is at working when substrate concentration. And the lower our Km, the better our
Working and
our enzyme is at working when substrate
lower substrate concentration. Working in lower substrate concentrations.
concentrations are small. And we can use this Km term to quantify an enzyme's ability to catalyze reactions, which we call catalytic efficiency. So I'll rewrite the Michaelis-Menten equation. Remember, we defined Km as a substrate concentration where VO is 1/2 Vmax. And since it's a concentration, it will be in units of molar or moles per liter. But now I'm going to throw a new term at you called Kcat, which is equal to the maximum speed of a reaction divided by the total enzyme available. And we call this the enzyme's turnover number. And all this term is how many substrates an enzyme can turn into product in 1 second at its maximum speed. And we measure it in units of seconds to the -1 or per second as in reactions per second. So we can define an enzyme's catalytic efficiency as a combination of Km and Kcat. And we do this by saying it's equal to Kcat over Km. So a higher Kcat or lower Km will result in an increase in an enzyme's catalytic efficiency. And every different enzyme has a different catalytic efficiency in certain conditions. And we can use this term to score enzymes on how good they are. So we covered a lot of content in this video, but the really crucial points to remember are first, the idea of the steady state assumption that we make when looking at enzyme kinetics. And this is where we assume that the ES concentration is constant, meaning that the formation and loss of ES are equal. Second, we derived the critically important Michaelis-Menten equation, which you should consider committing to memory. And third, we talked about how you can score how good an enzyme is at speeding up reactions by looking at that enzyme's catalytic efficiency, which is a combination of two new terms we learned about, KCAT, and an enzyme's ability to catalyze reactions.
Okay, we'll pause it here. We'll go over it, but I'm gonna take a break. Anybody with me? Nah, it's okay. All right, let's take a 10-minute, and we'll probably rewatch this one. I mean, we'll probably have to do a little bit more investigative work, but for now, let's at least rewatch that one. Let's go through this, continue my thing, and then we'll add it. We'll see based on high yield what it says. Might do a couple practice problems on this. Take a 10-minute. We're back. And we're back. Sorry, guys. It's from eating too much. You gotta go to the bathroom a lot, eh? Anyways, we're back. Let's continue with this. Hold on. Let me get a jacket. Yeah, that took a lot longer. Sorry about that, guys. Sorry about that. All right, we're back. We're back. Got the jacket. Not the jacket I wanted, but that's fine. Or hoodie, I guess.
Let's see. So I'll rewrite the Michaelis-Menten equation. Remember, we defined Km as a substrate concentration is 1/2 Vmax. And since it's a concentration, it will be in units of molar or moles per liter.
Hold on.
Substrate Km, which we call, is defined as the concentration of substrate at which our reaction speed is half of the Vmax. So when VO is equal to 1/2 of Vmax. And if we look at that on a graph from before, you'd see that Km is a substrate concentration specific to our circumstances, where our rate is at half of its max. And the lower our Km, the better our enzyme is at working when substrate concentrations are small. And we can use this Km term to quantify an enzyme's ability to catalyze reactions, which we call catalytic efficiency. So I'll rewrite the Michaelis-Menten equation. Remember, we defined Km as a substrate concentration, where VO is...
Okay, all right. Nice. It's speeding up. It's like he speeds up. Relax, buddy.
And we can use this Km term to quantify an enzyme's ability to catalyze reactions, which we call catalytic efficiency. And we can use this Km term to quantify an enzyme's ability to catalyze. As reactions, which we call catalytic efficiency. So I'll rewrite the Michaelis-Menten equation.
Yeah, you do that, buddy. You do that.
Remember, we defined Km as a substrate concentration where VO is 1/2 Vmax.
Mm-hmm.
And since it's a concentration, it will be in units of molar. Or moles per liter. But now I'm going to throw a new term at you called KCAT.
Throw them at me. Throw them at me. See if I catch them all.
Which is equal to the maximum speed of a reaction divided by the total enzyme available. The maximum speed of a reaction divided by the total enzyme available. And we call this the enzyme's turnover number. And all this term is how many substrates an enzyme can turn into product in 1 second. And all this term is how many an enzyme can
Turn into product
in 1
second at its maximum speed.
Is how many Substrates and enzyme can turn into product in 1 second at its maximum speed. And we measure it in units of seconds to this -1, or per second, as in reactions per second. So we can define an enzyme's catalytic efficiency as a combination of Km and Kk. Cat. And we do this by saying it's equal to Kcat over Km. So a higher Kcat or a lower Km would result in an increase in an enzyme's cat. Let's see. And every different enzyme has a different catalytic efficiency in certain conditions. And we can use this term to score enzymes on how good they are. So we covered a lot of content in this video, but the really crucial points to remember are first, the idea of the steady state assumption that we make when looking at enzymes. This is where we assume that the ES concentration is constant, meaning that the formation and loss of ES are equal. Second, we derived the critically important Michaelis-Menten equation, which you should consider committing to memory. And third, we talked about how you can score how good an enzyme is at speeding up reactions by looking at that enzyme's catalytic efficiency, which is a combination of two new terms we learned about, Kcat. and Km. So we're going to talk about cooperative binding, which is a very interesting topic when discussing enzyme kinetics. Divide enzyme catalysis into two steps. Create the enzyme, and second, the formation of product. And using this idea, we can derive the Michaelis-Menten equation, which is very useful for quantitatively looking at enzyme kinetics. Also remember that as you increase substrate concentration, the speed of product formation will level off at its maximum value, as shown on this graph. Now, the first thing that I want to talk about is that some proteins can bind more than one substrate, and not all enzymes have just one active site. So E can form ES through what I've called reaction 1, but can react with another molecule of substrate to form ES2 through what I've called reaction 2. And
ES2 plus S. 2 plus S.
again, to form ES3 through reaction 3, and so on. Now, these enzymes can form product at any stage of this process, no matter how many molecules of substrate are bound. Now, you would expect the rate of reaction 1 to be faster than the rate of reaction 2. If we're looking at the example of an enzyme with 3 substrate binding sites, there are 3 empty sites available for substrate to bind through reaction 1, and only 2 available for reaction 2, so you would expect rate 1 to be faster. Similarly, rate 2 would be faster than rate 3 for the same reason. And the idea is that the active site saturation does not increase with substrate concentration linearly. Now, I get that can be a mouthful, so let's...
Active site saturation does not increase linearly with an increase in the substrate concentration. Increase in the substrate concentration.
substrate concentration linearly. Now, I get that can be a mouthful, so let's look at it graphically. But we're also going to show an exception to this rule. So in this first graph, I've plotted substrate concentration against the percent saturation of the enzyme. And as you can see, the curve levels off as substrate binding sites become occupied. It becomes difficult to bind more substrate molecules as you have more substrate molecules bound. First graph, I've plotted substrate concentration against the percent saturation of the enzyme. And as you can see, the curve levels off as substrate binding sites become occupied. It becomes difficult to bind more substrate molecules as you have more substrate molecules bound. Next, I'm going to draw a different curve that you also might see in some enzymes, and that's where substrate binding happens more quickly as binding sites become occupied. Substrate binding changes substrate affinity, and we call this cooperativity. Now, with respect to cooperativity, we can define 3 new ideas. Positively cooperative binding occurs when substrate binding increases the enzyme's affinity for a subsequent substrate. Next, I'm going to draw a different curve that you also might see in some enzymes, and that's where substrate binding happens more quickly as binding sites become occupied. Substrate binding changes substrate affinity, and we call this cooperativity. Now, with respect to cooperativity, we can define 3 new ideas. Positively cooperative binding occurs when substrate binding
Substrate binding increases affinity for the subsequent substrate.
increases the enzyme's affinity for a subsequent
It's called
substrate.
positively cooperative.
Negatively cooperative binding occurs when substrate binding decreases the enzyme's affinity for subsequent substrate more than you would normally expect, and non-cooperative binding is the same as the first example, where substrate binding does not affect the enzyme's affinity for substrate molecules. So let's look at this graphically.
Okay.
If we have a protein with, let's say, 5 binding sites, and plot the fraction occupied versus the substrate concentration, we would come up with 3 possible curves. The green curve, which takes on a sigmoidal shape, would represent an enzyme with positive cooperativity. The blue curve with a hyperbolic shape would represent an enzyme with non-cooperative binding. And the red curve would represent an enzyme with negatively cooperative binding. Now remember that the effects of cooperativity are only seen after some substrate has already bound, which is why the difference in the fraction occupied between the 3 curves is much smaller at smaller values, like the 1/5 that I've shown here, than it is at the higher values. So let's look at a specific example of a couple proteins. So hemoglobin, or Hb, is the oxygen-carrying molecule that you find in human blood, and it can bind up to a total of 4 oxygen molecules, and it exhibits positively cooperative binding. Myoglobin, on the other hand, which is the oxygen-carrying molecule that you find in muscle tissue, can only bind 1 oxygen molecule in total. And since it can only bind 1, it must exhibit non-cooperative binding, since there's no subsequent substrate to speak of. Now, if we make a graph where we plot the fraction of active sites bound in each of these proteins versus the pressure of oxygen, remember oxygen is our substrate here, and since it's a gas, we're going to use pressure instead of concentration. You can see that the red sigmoidal curve associated with hemoglobin's positive cooperative binding looks different from the blue hyperbolic curve associated with myoglobin's non-cooperative binding. So what did we learn? Well, first we learned that some proteins can bind more than 1 non-cooperative binding. So what did we learn? Well, next we learned that There are three different types of cooperativity: positive, negative, and non-cooperative. Finally, we learned about proteins that exhibit two different types of cooperativity, which are the oxygen-binding molecules hemoglobin and myoglobin. So today we're going to talk about how allosteric regulation can affect enzyme kinetics. But first, let's review the idea that an enzyme's catalysis can be divided into two steps. First, the binding of enzymes to substrate, and second, the formation of products. And using this information, we can derive the Michaelis-Menten equation, which allows us to look at an enzyme's rate of product formation with respect to substrate concentration. Also remember that substrates will typically bind to enzymes at the active site. So what do we mean when we say allosteric regulation? Well, we know that enzymes usually have an active site where substrates can bind, but enzymes can also have what we call an allosteric site. And these allosteric sites are places on the enzyme where any enzyme regulator can bind. And I've put this star here just to point out that allosteric sites can be anywhere on an enzyme, and there can be any number of them as well. So what do we mean when we say regulators? Well, we generally say that there are two types of regulators. There are allosteric activators, which increase enzymatic activity and activate them, and allosteric inhibitors, which decrease enzymatic activity and inhibit the enzymes. So let's take a look at what we mean by increasing and decreasing enzymatic activity from a kinetic perspective. So remember the Michaelis-Menten equation, and if we're assuming substrate concentration to be constant, then there are two ways to influence enzymatic activity, or VO. And in this first graph, I've drawn three different curves, and the blue curve represents the enzyme functioning without an allosteric regulator at all. The red curve represents an enzyme with an allosteric inhibitor, and the green curve represents the enzyme with an allosteric activator. And in this example, the activators and inhibitors affect VO through either increasing or decreasing Km, since the Vmax values seem to be pretty close between the three curves. So an activator here might be decreasing Km. Now in this next example, we have the same three colored curves, but instead of Km changing significantly, the regulators seem to be changing Vmax, with the activator in. First, let's review the idea that an enzyme's catalysis can be divided into two steps. First, the binding of enzymes to substrate, and second, the formation of products. And using this information, we can derive the Michaelis-Menten equation, which allows us to look at an enzyme's rate of product formation with respect to substrate concentration. Also remember that substrates will typically bind to enzymes at the active site. So what do we mean when we say allosteric regulation? Well, we know that enzymes usually have an active site where substrates can bind, but enzymes can also have what we call an allosteric site. And these allosteric sites are places on the enzyme where any enzyme regulator can bind. And I've put this star here just to point out that allosteric sites can be anywhere on an enzyme, and there can be any number of them as well. So what do we mean when we say regulators? Well, we generally say that there are two types of regulators. There are allosteric activators, which increase enzymatic activity and activate them, and allosteric inhibitors, which decrease enzymatic activity and inhibit the enzymes. So let's take a look at what we mean by increasing and decreasing enzymatic activity from a kinetic perspective. So remember the Michaelis-Menten equation, and if we're assuming substrate concentration to be constant, then there are two ways to influence enzymatic activity, or VO. And in this first graph, I've drawn three different curves, and the blue curve represents the enzyme functioning without an allosteric regulator at all. The red curve represents an enzyme with an allosteric inhibitor, and the green curve represents the enzyme with an allosteric activator. And in this example, the activators and inhibitors affect VO through either increasing or decreasing Km, since the Vmax values seem to be pretty close between the three curves. So an activator here might be decreasing Km. Now in this next example, we have the same three colored curves, but instead of Km changing significantly, the regulators seem to be changing Vmax, with the activator increasing the Vmax value. So now that we've talked about activators and inhibitors, let's introduce the idea of the feedback loop. And the basic idea is that a feedback loop is when you have downstream products regulating upstream reactions. And I understand this can be a mouthful, so let me show you this little reaction sequence where we have A forming B through reaction 1, and B forming C through reaction 2, and so on and so on. Now let's say that molecule F acted as an activator for the enzyme powering reaction 1. So it had a positive effect on enzyme 1's activity. Now we would call this a positive feedback loop since molecule F increases the rate of reaction 1, which then causes even more F to be made since we've increased the rate of formation of molecule F. Now let's say molecule F had a negative effect on enzyme 1. We would call this a negative feedback loop since molecule F decreases the rate of reaction 1, which leads to a decrease in the rate of formation of molecule F. So let's look at an example of a feedback loop, just to really drive home the point if you're still confused. Now, phosphofructokinase is an enzyme involved in glycolysis, and it catalyzes the conversion of fructose 6-phosphate and ATP to form fructose 1,6-bisphosphate and ADP. Now remember that glycolysis is a metabolic process that cells use to generate ATP. So here our molecule F, or downstream regulator from the last example, is ATP. And it turns out that ATP is an allosteric inhibitor of phosphofructokinase. And this makes sense because if ATP is at a high level, then it's like the cell saying, we have ATP and we don't really need any more. And we don't need phosphofructokinase to push glycolysis along. So this would be a good example of a negative feedback loop, since making ATP slows down glycolysis and thus slows down the rate of ATP production. Now, because ATP is both an allosteric regulator and a substrate for phosphofructokinase, we can call it a homotropic inhibitor, which is a new term, and we call it a homotropic inhibitor because the substrate and the regulator are the same molecule. Now, AMP, which is used up ATP, is an activator for phosphofructokinase, and this also makes sense because if AMP levels are high, then ATP levels are probably low, and it's like the cell saying, we need ATP. So we do need phosphofructokinase to push glycolysis along. Now, since AMP is a regulating molecule but not an active site substrate for phosphofructokinase, it would be considered a heterotropic activator, since the substrate and regulator are different. Now, the final point I want to make is that specific reactions make excellent control points for long multi-step processes. And remember that glycolysis is a 10-step sequence. So why is there so much regulation going on for this one step? Well, this reaction in particular has a varying regulator are different. Now, the final point I want to make is that specific reactions make excellent control points for long multi-step processes. And remember that glycolysis is a 10-step sequence. So why is there so much regulation going on for this one step? Well, this reaction in particular has a very negative delta G, and it's actually negative 4.5 kcal per mole. And that means that it's not easily reversed since there will be a big release of energy from the reaction. And this makes this step of glycolysis an excellent control point for all 10 steps together, since it's more or less a one-way reaction. So what did we learn? Well, first we learned about glycolysis, an excellent control point for all 10 steps together, since it's more or less a one-way reaction. So what did we learn? Well, first we learned about the concept of allostery and how regulatory molecules can bind to allosteric sites instead of active sites. Second, we learned that these allosteric regulators influence an enzyme's kinetics by increasing or decreasing Km or Vmax. And third, we learned about what a feedback loop is and how in long multi-step processes like glycolysis, the best control points are highly committing steps, the ones with very negative delta G values. In long multi-step processes like glycolysis, The best control points are highly committing steps, the ones with very negative delta G values. So today we're going to learn about covalent modifications to enzymes. But first, let's review the idea that enzymes make reactions go faster. And looking at a reaction coordinate diagram, you'd notice that enzymes do this by lowering the reaction's activation energy. Also, before we talk about covalently modified enzymes, I want to remind you that not all enzymes are proteins. And often when we think of enzymes, we think of proteins, which are amino acid polymers with primary, secondary, tertiary, and quaternary structures. But there are also many different kinds of enzymes that aren't proteins. Inorganic metals like magnesium or small organic molecules like flavin can also act as enzymes. But for the purposes of this discussion, we're going to focus on the proteins. And to be clear, when we say covalent modifications, we refer to modifications to a protein that involve forming or breaking covalent bonds. Now, there are tons of different covalent modifications that we can observe, so I'm only going to touch on a select few to get the point across. And the first category of covalent modifications I want to talk about are small post-translational modifications. Now, when I say translation, I'm referring to the process of translation, where amino acid polymers are synthesized. When I say post-translation, I refer to events that take place after that initial synthesis. Now, when I say small, all I'm referring to are modifications that involve small functional groups being added or removed from an enzyme. And again, there are many different types of these, but I'm just going to touch on three. So methylation is a modification of a protein that involves the addition of a methyl group, or CH3, to a protein. Acetylation involves the addition of an acetyl group, and glycosylation involves the addition of a sugar molecule. And these are just three examples of a huge list. And these modifications, although small, can have pretty significant impacts on the protein as a whole. And to discuss this, I want to mention the example of acetylation of a lysine residue on a protein. So as you may know, lysine is an amino acid that has an extra amino group on its side chain that can act as a base and carry a positive charge. If we were to acetylate this lysine residue and add an acetyl group to the amino nitrogen, which is a covalent modification, the electron withdrawing effect of the acetyl group prevent that nitrogen from carrying a positive charge and modify the behavior of that amino acid. The loss of that positive charge can change a few properties of the amino acid, including changes to the lysine's acidity and basicity, exchange protons as well, and it'll also influence lysine's electrostatic interactions with other charged molecules since it's lost that positive charge. So even a small modification. Moving on, I want to discuss another way a acetyl group can have significant impact. Moving on, I want to discuss another way in which covalent modifications of enzymes is relevant, and that's in reference to zymogens. Now, a zymogen in order to become active. And a big example of these in biology are the digestive enzymes that the pancreas releases so that you can digest food. One of the enzymes that the pancreas releases is called trypsinogen, which is a zymogen, as indicated by the ogen suffix. Now, this is an inactive form of a protease enzyme that is shipped to the intestine. And once in the intestine, it's covalently modified by an enzyme called enterokinase, which converts it to its active form, trypsin. Now, this is to prevent trypsin from breaking down proteins that we need in the pancreas, since it's inactive at that point as trypsinogen, and only allows it to break proteins down in the intestine after it's encountered enterokinase. Notice how you can distinguish zymogens from their active form by their name, in that zymogens have ogen added to the end of them. Now, the last example of covalently modified in that zymogens have ogen added to the end of them. Now, the last example of covalently modified enzymes that I want to discuss is the subject of suicide inhibition. Now, when we think of enzymatic inhibition, we usually think of competitive, non-competitive, and uncompetitive inhibitors, which follow certain patterns in terms of their effects on enzyme kinetics. But there's another type of inhibitor that's a little different, and this is the suicide inhibitor. So that since these inhibitors have covalent linkages to the proteins, they rarely unbind, which is why we call them suicide inhibitors, since after they bind, that's it for them. And this is what distinguishes this type of inhibitor from the other three that you might be familiar with. So what did we learn? Well, we talked about three very different things today that all have to do with covalent modifications to enzyme. First, we talked about small post-translational modifications like methylation, acetylation, and glycosylation. Second, we discussed zymogens, inactive proteins that require covalent modification to become active. And finally, we talked about suicide inhibitors, which are enzyme inhibitors that permanently bind their target.
Yeah, that makes sense. Cofactors. Maybe we can watch these ones too. baby. Nice stuff, man. Ah. I think we should get some. Should I take another B12? The limit? Maybe not. I don't think so. Won't build up too much tolerance. Yeah, let's stay on it, I guess. What happened? Yeah, team control. Okay. Just take it off. Take it off for now. I think we're okay. We're just. Let's work out after this, though. I need some blood, you know? We'll take a 15 and then work out before we start the next ones. We got 3 to do, technically. 1, 2, 3. Well, I'll just quickly go over these. Wait, oh no. Shoot, I'm on the wrong one. What the heck? Where is this one? Wait, what? It's the same one. Oh, okay. Is this it? Yeah, it's the same one. Why is it 2? Why was it duplicate? Oh my goodness. Same. Oh, no inhibition. K is 20. Oh yeah, less substrate is needed. More substrate is needed. But it has the same Vmax. Less substrate is needed. Mm. Is that always the case? Oh, non-competitive inhibitor. Uncompetitive inhibitor. What the heck? Jesus, man. Like, if you're gonna do it, give me the right information. What? does not bind to the active site.
about enzymes. And what I want to do in this video is dig a little bit deeper and focus on some actors that actually help enzymes. Just as a reminder, enzymes are around to help reactions to proceed, to lower their activation energies, to make the reactions happen more frequently or to happen faster. Now, we've already seen examples of enzymes. It's just to frame things in our brain properly. Sometimes in a textbook, you'll see an enzyme like this. You see a drawing like this, and people will call this the enzyme. They'll call this the enzyme, and then they'll call this, right? They'll say, okay, and it's acting on some type of a substrate right over here. It's going to do something to that. And this is nice for a very abstract textbook idea of a substrate locking into an enzyme like this, but this isn't actually what it looks like in a biological system. We have to remind ourselves, when people talk about enzymes, they're talking about proteins. Now, there are these kind of RNA enzymes called ribozymes, but the great majority, when we're talking about enzymes, we tend to be talking about proteins. And we spend a lot of time talking about how proteins are these structures, there's polypeptides and all the side chains of the various amino acids hold the proteins in all sorts of different ways. So if I were a better drawing for something like this, it would be this protein. It's all folded in different ways. Maybe it has some alpha helices here. Maybe it has some beta sheets right over here. It's all this kind of crazy stuff right over there. And then the substrate might be some type of a molecule. That is, it gets embedded. It's embedded in the protein. And you see some examples right over here. This is actually a hexokinase model, and you see at least you can see a little bit of the ATP right over there, and it's a little harder to see the glucose that's going to be phosphorylated, and this reaction is being facilitated by this big protein structure, the hexokinase. Now, what we're going to focus on in this video is that when we talk about an enzyme, we're talking about proteins. We're talking about a chain of amino acids. But there's often other parts of the enzyme that aren't officially proteins. And we even saw that when we talked about hexokinases, when we talked about the phosphorylation of glucose. We said, hey, the way that it lowers the activation energy is you have these positive magnesium ions. These positive magnesium ions that can keep the electrons in the phosphate groups a little bit busy, draw them away, so that this hydroxyl group right over here can bond with this phosphate and not be interfered with these electrons. Well, these magnesium ions right over here, they aren't officially part of the protein. These are what we call cofactors. So you might have a cofactor right over there that latches on to the broader protein to become part of the enzyme, and you actually need that for the reaction to proceed. It plays a crucial role here. So another drawing in the textbook, you'll see something like this, or even they'll say, okay, in order for this reaction to proceed, yes, you need the substrate, but you also need the cofactor. The cofactor. And once again, it sounds like a fancy word, but all it means is the non-protein part of an enzyme. It's another molecule or ion or atom that is involved in the enzyme performance function, that it's not formally part of an amino acid or part of a side chain or part of the protein. But it's another thing that needs to be there to help catalyze the reaction. We saw that with hexokinase. You had magnesium ions that the complex picks up. And this is why, when people talk about your vitamins and minerals, a lot of the vitamins and minerals that you need, they actually act as cofactors for enzymes. And so you can even see it in this drawing over here, at least based on what I read. These are the magnesium ions in green right over here, and these are cofactors. These are cofactors. So cofactor, non-protein part of your actual enzyme. Now we can subdivide cofactors even more. We can divide them into organic cofactors and or inorganic cofactors. You have cofactors. We've seen an inorganic cofactor. A lot of these ions, you see magnesium ions, you see sodium ions, you see calcium ions, you see all sorts of things acting as cofactors, oftentimes to distract electrons or to keep them busy so that electrons can proceed. But you can also have organic ones. You can also have organic molecules. Remember, organic molecules, they'll involve carbon. They have chains of carbons and other things. And cofactors that are organic molecules, we call them coenzymes. Coenzymes. And there's a bunch of examples of coenzymes. This right over here is the enzyme lactate dehydrogenase, and it has the coenzyme. The coenzyme you are going to see a lot in your biological careers, NAD. Notice this isn't just an ion. It is an entire molecule. It has carbon in it. That's why we call it organic. And it is not formally protein. It's not part of the amino acids that make up the protein. So that's what makes it a cofactor. And since it's an entire organic molecule, we call this a coenzyme. Coenzyme. But like any cofactor, it plays a role in actually allowing the enzyme to do its function, to facilitate. It is an entire ions. We'll see calcium ions. We'll see all sorts of things acting as cofactors, oftentimes to distract electrons so that, or to keep them busy so that electrons can proceed. But you can also have organic ones. You can also have organic molecules. Remember, organic molecules, they'll involve carbon. They have chains of carbons and other things. And cofactors that are organic molecules, we call them coenzymes. Coenzymes. And there's a bunch of examples of coenzymes. This right over here is the enzyme lactate dehydrogenase, and it has the coenzyme, and this coenzyme you are going to see a lot in your biological careers, NAD. This isn't just an ion. It is an entire molecule. It has carbon in it. That's why we call it organic. And it is not formally protein. It's not part of the amino acids that make up the protein. So that's what makes it a cofactor. And since it's an entire organic molecule, we call this a coenzyme. Coenzyme. But like any cofactor, it plays a role in actually allowing the enzyme to do its function, to facilitate reaction. And this particular coenzyme, NAD, which you're going to see a lot, it helps facilitate the transfer of hydride ions. Hydride ions never or very seldom exist by themselves, but it's a hydrogen with an extra electron, so it has a negative charge. So it allows the transfer of this group or from a substrate or to a substrate, and that's because NAD can accept a hydride anion right over here and become NADH. And if you want to see its broader structure, it's actually quite fascinating. I'll probably do a whole video on NAD because in so many textbooks growing up, I just saw, NAD and NADH, and I'm like, what is this thing? And it's a fascinating molecule. So what it can do is it can actually pick up the hydride anion right over here at this carbon. You can actually form another bond with the hydrogen. I'll do that in a future video. I'll show the mechanism for it. But it's a pretty cool molecule, and I like to actually look at this molecule. Remember, the whole focus of this is coenzymes, but we see these patterns throughout biology because the name nicotinamide adenine dinucleotide exactly describes what it is. Nicotinamide, right down here, that is this piece of the molecule, and this is the part that can accept a hydride or let go of a hydride. So you can say this is the active part of the molecule. Adenine, our good old friends. We've seen adenine in DNA and RNA and ATP. So this is our good old friend adenine right over here. And it says dinucleotide because we actually have two nucleotides paired together. Their phosphate groups are tied together. And there's a couple of cool ways to think about this. You have an adenine right over here. You have a ribose. You have a phosphate group. If you just looked at this piece right over here, if you look at this right over here, this is your building block. This is your building block of or this could be a building block of RNA if you have an adenine right over there. And if you include all of this right over here, this is ADP. The reason why it's called dinucleotide, you can also divide it the other way. You can say, all right, you have one nucleotide that has nicotinamide right over here. So that's one of the nucleotides, and then the other nucleotide is right over here, the one that involves adenine. That's what's called dinucleotide. So hopefully this makes NAD less of a mysterious molecule. We'll see it in the future. But I like to look at it because it's got all these patterns, it's got all these components you see over and over again. You see it in ATP, you see it in RNA over and over again. But this isn't the only cofactor or coenzyme. There are many others. In fact, when people say take your vitamins and your minerals, that tends to be because they are cofactors. Vitamin C is a very important cofactor to be involved in enzymes. Well, all the different things that can do. These are two different views of vitamin C. It's a base-building model. This is a ball and stick model right over here, vitamin C. Folic acid, once again, two different views. But these are all coenzymes. They all work. If you have a protein right over here, then it's all this really complex structure. Maybe you have some substrates, but to help facilitate the substrates in different colors, maybe you have some as in the mechanism charge. Sometimes it might be a series of different as part of the reaction mechanism. We've already seen that an enzyme helps catalyze a reaction. So let's say this right over here, this is our enzyme, and we have our substrate, and it goes and it binds to the active site, to the active site of the enzyme. So let's say it binds right over there. So that site on the enzyme we call the active site where the substrate binds, and then the enzyme catalyzes a reaction. Maybe it breaks up this substrate into two smaller molecules. And so after the reaction, after the reaction, the enzyme is unchanged, but a reaction has been catalyzed. We now have the substrate being broken up, in this case at least, into two smaller molecules. Maybe I'll draw them. That's one of them, and this is the other one right over here. So they just came from the active site. Once the reaction is catalyzed, they don't have the affinity to the active site anymore, and they break off. So this enzyme has just catalyzed this reaction. What I want to talk about in this video is how this might be inhibited, and specifically how it might be inhibited competitively. So we're going to talk about competitive inhibition. So competitive, right over here, competitive inhibition. So the classic case of competitive inhibition, if there's some molecule that competes for the substrate at the active site. As we'll see, this isn't the only form of competitive inhibition, but this is the one that you will most typically see in a textbook. So so that's our enzyme again. And so that's our enzyme. And we've already seen that this is this is right over here where I'm circling, that is the active site. Active site. And if the molecule, the intended substrate, I guess you could say, gets to it, we're going to have this first scenario up here. But in classic competitive inhibition, or at least the version I'm just going to show you right now, you could have another molecule. That, it looks something like this, that can compete for the active site. And if it gets to the active site first, so if it gets there first, so let me show what's going to happen. So we have our enzyme, we have the other molecule, not the intended substrate, binds to the active site first. Well, now the intended substrate, the one for which the enzyme catalyzes a reaction, isn't able to bind, and the reaction isn't going to happen. And you can see very clearly that they're competing for the enzyme, and in this case, they're competing for the active site. Now, this isn't the only form of competitive inhibition. Another form of competitive inhibition is allosteric competitive inhibition. Let me write this down. For the active site, and if it gets to the active site first, so if it gets there first, so let me show what's going to happen. So then we have our enzyme, we have the other molecule, not the intended substrate, binds to the active site first. Well, now the intended substrate, the one for which the enzyme catalyzes a reaction, isn't able to bind, and the reaction isn't going to happen. And you can see very clearly that they're competing for the enzyme, and in this case, they're competing for the active site. Now, this isn't the only form of competitive inhibition. Another form of competitive inhibition is allosteric competitive inhibition. Let me write this down. So you have allosteric competitive inhibition. Inhibition. And an allosteric site is a site other than the active site. But in allosteric competitive inhibition, or competitive allosteric inhibition, however you want to say it, you have a scenario where the competitor doesn't bind to the active site, but binds to a site that is not the active site, an allosteric site, you could say. So in that one, the competitor maybe might bind here. So that's clearly not the active site. So maybe the competitor looks something like that. It didn't bind to the active site, but by binding there, the active site can no longer bind to the intended substrate. So you have the same effect. You have the same effect right over here, where this thing isn't going to bind. But if this thing binds first, let me draw that scenario. So if the intended substrate binds first, then the competitor can't bind. So in this scenario, if the substrate is able to get to the active site, well, then the competitor can bind. So once again, they're competing. So I'll draw the competitor up here. So then the competitor, whoever gets to it first gets the enzyme. So in this situation, the competitor is not going to bind. So that's true of whether you're talking about competitive inhibition where they're competing for the active site. If the competitor gets there first, the intended substrate isn't going to get there. The reaction isn't going to be catalyzed. If the intended substrate gets first, then the competitor is not going to be able to get there. In fact, it could have been this situation where because the substrate got there first, the competitor isn't going to be able to bind to the active site. When we're talking about allosteric competitive inhibition, we're still competing for the enzyme. Only one's going to get it. If one gets to the enzyme first, then the other one's not going to be able to get there. They are competing for the enzyme, but the competitor, the non-substrate, is just acting at an allosteric site. By binding to an allosteric site, it changes the conformation of the enzyme so that the active site no longer binds to the substrate. And I want to really emphasize this point because when I first learned this, I said, and it's often sometimes confused, and even some things you'll read on the internet, that they'll say that this allosteric type of inhibition, they'll call this non-competitive because you're not competing for the active site. But that is actually not the case. In non-competitive inhibition, and I'm going to do the whole next video on non-competitive inhibition. In non-competitive inhibition, the inhibitor right over here, can bind regardless of whether the substrate has bound or not. But when the inhibitor does bind, it prevents the reaction from moving forward. It changes the conformation of the protein so it no longer catalyzes the reaction. So non-competitive, they both can bind, but if the inhibitor is there, the reaction isn't going to proceed. In competitive inhibition, whether we're talking about allosteric or non-allosteric competitive inhibition, only one of the substrate or the inhibitor is going to be able to bind. They're competing for the enzyme. In the video on competitive inhibition, we saw that competitive inhibition is all about a substrate or potential substrate, an inhibitor competing for the enzyme. And whoever gets there first gets the enzyme. If the inhibitor gets there first, then the substrate isn't able to bind, and of course, no reaction is catalyzed. If the substrate is able to get there first, then the inhibitor isn't able to bind, and the reaction does get catalyzed. Now, the inhibitor and the substrate, they both might compete for the active site, if we're talking about competitive inhibition, but you also have allosteric competitive inhibition, where they're still trying to compete for the enzyme. Whoever gets there first gets the enzyme, but the inhibitor doesn't necessarily bind at the active site. They bind at an allosteric site, but it's the same idea. If the inhibitor gets to the allosteric site before the substrate gets to the active site, then the conformation of the protein changes so that the active site, it changes a little bit. We try that same color, changes the conformation of the protein, changes a little bit, and then let's draw that. And so can an inhibitor. And the inhibitor can bind at an allosteric site. So this is we kept talking about it a little bit. So of course the substrate can bind, and so can an inhibitor. So now the reaction is going to look like this. If this happens, the only option is that they both unbind. So now this character is just going to leave the active site. No reaction has been catalyzed. So this prevented anything from happening. Maybe this guy leaves as well. And the way I showed this non-competitive inhibition, I showed it happening at an allosteric site, the inhibitor attaching at an allosteric site, but it actually doesn't even have to be the same case as long as it does not prevent as long as it can actually bind close to or even at the active site, as long as it does not prevent the substrate from binding to the active site. So you can even have a situation like this. This is the one that's typically given for non-competitive inhibition, where you have the inhibitor binding in an allosteric site. But the idea here is that they're not competing. Both of them can bind to the enzyme. If one of them binds first, then the other one can still bind. If the substrate binds first, then the inhibitor can still bind. If the inhibitor binds first, then the substrate can still bind. But if the reaction is not going to be catalyzed. But you can even have a situation.
Well, the reactions are going to be catalyzed. They could still occur. Right?
You can even have a situation where the inhibitor and the substrate can both bind in or around the active site. So that's the inhibitor. And then this is our substrate. But once again, this reaction is not going to occur. We have non-competitive inhibition. They're not competing for the thing. They can both bind to it. Whether they can bind is dependent on whether the other one is bound. But if the inhibitor is there, then it's not going to allow the reaction to actually be catalyzed. As opposed to competitive inhibition, whoever gets to the enzyme first gets the enzyme. Hopefully that clarifies things.
Mm-hmm. This would be allosteric non-competitive inhibition, right? Allosteric non-competitive. Okay.
As opposed to competitive inhibition, whoever gets that clarifies it. About how the hemoglobin or the red blood cells, depending on what level you want to operate, know, and I have to use know in quotes, these aren't sentient beings, but how do they know when to pick up the oxygen and when to drop off the oxygen? So this right here, this is actually a picture of a hemoglobin bin. And if you total up the hemoglobin in the entire body, it's huge because it's been reacted with. That can also actually shows the cooperative binding. So let's say we're just dealing a lot about how the hemoglobin or the red blood cells, depending on what level you want to operate, know, and I have to use know in quotes, these aren't sentient beings, but how do they know when to pick up the oxygen and when to drop off the oxygen? So this right here, this is actually a picture of a hemoglobin protein right there. This is hemoglobin. It's made up of 4 amino acid chains. That's one of them. Those are the other 2. We're not going to go into the detail of that, but these look like little curly ribbons. If you imagine them, they're a bunch of molecules and amino acids, and then they're curled around like that. So this, on some level, describes its shape. And then in each of those groups, or in each of those chains, you have a heme group. You have a heme group here in green. Heme group. That's where you get the heme and hemoglobin from. You have 4 heme groups, and the globins are essentially describing the rest of it, the protein structures, the 4 peptide chains. Now, this heme group, this is pretty interesting. It actually is a porphyrin structure, and if you watch the video on chlorophyll, you remember a porphyrin structure. But at the very center of it, in chlorophyll, we had a magnesium ion. But at the very center of hemoglobin, we have an iron. We have an iron ion. And this is where the oxygen binds. So on this hemoglobin, you have 4 major binding sites for oxygen. You have right there, maybe right there, a little bit behind, right there, and right there. Now, why is hemoglobin? Well, oxygen will bind very well here. But hemoglobin has several properties that, one, make it really good at binding oxygen, and then also really good at dumping oxygen when it needs to dump oxygen. So it exhibits something called cooperative binding. Cooperative binding. And this is just the principle that once it binds to one oxygen molecule, so let's say one oxygen molecule binds right there, it changes the shape in such a way that the other sites are more likely to bind oxygen. So it just makes it, one binding makes the other bindings more likely. Makes other bindings more likely. Now you say, okay, that's fine. That makes it a very good oxygen acceptor when it's traveling through the pulmonary capillaries, and oxygen is diffusing from the alveoli. That makes it really good at picking up the oxygen. But how does it know when to dump the oxygen? This is an interesting question. When to dump oxygen? It doesn't have eyes or some type of GPS system that says, oh, you know what? This guy is running right now, and so he's generating a lot of carbon dioxide right now in these capillaries, and he needs a lot of oxygen in these capillaries surrounding his quadriceps. I need to deliver oxygen. It doesn't know it's in the quadriceps. How does the hemoglobin know to let go of the oxygen there? And that's a byproduct of what we call allosteric inhibition, which is a very fancy word, but the concept's actually pretty straightforward. Allosteric inhibition. And when you talk about allosteric anything, it's often used in the context of enzymes. You're talking about the idea that things bind to other parts. Allo means other. So you're binding to other parts of the protein or the enzyme, and enzymes are just proteins, and it affects the ability of the protein or the enzyme to do what it normally does. So hemoglobin is allosterically inhibited by carbon dioxide, and by protons. So carbon dioxide can bond to other parts of the hemoglobin, I don't know the exact spots, and so can protons. So remember, acidity just means a high concentration of protons. So if you're in an acidic environment, protons can bond. Maybe I'll do the protons in this pink color. Protons, which are just hydrogen without electrons, right? Protons can bond to certain parts of our protein, and it makes it harder for them to hold on to the oxygen. So when you're in the presence of a lot of carbon dioxide or an acidic environment, this thing is going to let go of its oxygen. And it just happens to be that that's a really good time to let go of your oxygen. Let's go back to this guy running. He's running. There's a lot of activity in these cells right here in his quadriceps. They're releasing a lot of carbon dioxide into the capillaries, into well, at that point they're going from arteries into veins, and they need a lot of oxygen, which is a great time for the hemoglobin to dump their oxygen. So it's really good that hemoglobin is allosterically inhibited by carbon dioxide. Carbon dioxide means on certain parts of it, it starts letting go of its oxygen. That's exactly
Allosterically inhibited.
where in the body the oxygen is needed. Now you're saying, wait, what about this acidic environment? How does this come into play? Well, it turns out that most of the carbon dioxide is actually disassociated. It actually disassociates. It does go into the plasma, but it actually gets turned into carbonic acid. So I'll just write a little formula right here. So if you have a little bit of, if you have some CO2 and you mix it with the water, I mean, most of our blood, the plasma, it's water. So you take some carbon dioxide, you mix it with water, and you have it in the presence of an enzyme, and this enzyme exists in red blood cells. It's called carbonic anhydrase. Carbonic anhydrase. A reaction will occur that'll essentially you'll end up.
Hold on, Mom. Hold on.
Up with carbonic acid. You'll end up with, we have H2CO3. It's all balanced. We have 3 oxygens, 2 hydrogens, 1 carbon. And this it's called carbonic acid because it gives away hydrogen protons very easily. Acids dissociate into their conjugate base and hydrogen protons very easily. So carbonic acid disassociates very easily. It's an acid, although I'll write in some type of an equilibrium right there. If any of this notation really confuses you, if you want more detail on it, watch some of the chemistry videos on acid dissociation and equilibrium reactions and all of that. But it essentially can give away one of these hydrogens. So you're left with, but just the proton, and it keeps the electron of that hydrogen. So you're left with a hydrogen proton plus, well, you gave away one of the hydrogens, so you just have 1 hydrogen. This is actually by a bicarbonate ion. So this is bicarbonate right there, but it only gave away the proton, kept the electron, so you have a minus sign. So it's the all of the charge adds up to neutral, and that's neutral over there. So if I'm in a capillary of the leg, let me see if I can draw this. So let's say I'm in the capillary of my leg. Let me do a neutral color. So this is a capillary of my leg. I've zoomed in just one part of the capillary, and it's always branching off. And over here, I have a bunch of muscle cells right here that are generating a lot of carbon dioxide. They're generating a lot of carbon dioxide, and they need oxygen. Well, what's going to happen? Well, I have my red blood cells flowing along. It's actually interesting. Red blood cells, that the are 25%, their diameter is 25% larger than the smallest capillaries. So essentially, they get squeezed as they go through the small capillaries, which a lot of people believe helps them release their contents. So releases maybe some of the oxygen that they have in them. So you have a red blood cell that's coming in here. It's being squeezed through this capillary right here. It has a bunch of hemoglobin. And when I say a bunch, you might as well know right now, each red blood cell has 270 million hemoglobin proteins, 270 million. And if you total up the hemoglobin in the entire body, it's huge because we have, let me write this number down. We have 20 to 30 trillion red blood cells. We have 20 to 30 trillion red blood cells, and each of those 20 to 30 trillion red blood cells have 270 million hemoglobin proteins in them. So we have a lot of hemoglobin. So anyway, that was a little bit of a, so actually red blood cells make up roughly 25% of all of the cells in our body. We have about 100 trillion or a little bit more, give or take. I've never sat down and counted them. But anyway, we have 270 million hemoglobin particles or proteins in each red blood cell. Explains why the red blood cells had to shed their nucleuses to make space for all of those hemoglobins. They're carrying oxygen. So right here we're dealing with, this is an artery, right? It's coming from the heart. The red blood cell is going in that direction, and then it's going to shed its oxygen, and then it's going to become a vein. Now, what's going to happen is you have this carbon dioxide, you have a high concentration of carbon dioxide in the muscle cell, and eventually, just by diffusion gradient, ends up, let me do that same color, ends up in the blood plasma, just like that. And some of it can make its way across the membrane into the actual red blood cell. In the red blood cell, you have this carbonic anhydrase, which makes the carbon dioxide dissociate into, or essentially become carbonic acid, which then can release protons. So let me do the protons out here. Well, those protons, we just learned, can allosterically inhibit the uptake of oxygen by hemoglobin. So those protons start bonding to different parts, and even the carbon dioxide that hasn't been reacted with, that can also allosterically inhibit the hemoglobin, so it also bonds to other parts, and that changes the shape of the hemoglobin protein just enough that
Oh, what? Ah.
it can't hold on to its oxygens that well, and it starts letting go. And just as we said, we had co-opted binding. The more oxygen
you have on, the better it is at accepting more. And that changes the shape of the hemoglobin protein just enough that it can't hold on to its oxygens that well. It starts letting go. And just as we said, we had cooperative binding. The more oxygens you have on, the better it is at accepting more. The opposite happens. When you start letting go of oxygen, it becomes harder to retain the other ones. So then all of the oxygens let go. So this is, at least in my mind, it's a brilliant mechanism, because the oxygen gets let go just where it needs to let go. It doesn't just say, oh, I've left an artery and I'm now in a vein. Maybe I've gone through some capillaries right here and I'm gonna go back to a vein. Let me release my oxygen, because then it would just release the oxygen willy-nilly throughout the body. This system, by being allosterically inhibited by carbon dioxide and an acidic environment, it allows it to release it where it is most needed, where there's the most carbon dioxide, where respiration is occurring most rigorously, or vigorously. So it's a fascinating scheme. And just to get a better understanding of it, right here I have this little chart right here that shows the oxygen uptake by hemoglobin, or how saturated it can be. And you might see this in maybe your biology class. It's a good thing to understand. So right here we have on the x-axis, or the horizontal axis, we have the partial pressure of oxygen. And if you watch the chemistry lectures on partial pressure, you know that partial pressure just means how frequently are you being bumped into by oxygen. Pressure is generated by gases or molecules bumping into you, or it doesn't have to be gas, but just molecules bumping into you. And then the partial pressure of oxygen is the amount of that that's generated by oxygen molecules bumping into you. So you can imagine as you go to the right, there's just more and more oxygen around, so you're going to get more and more bumped into by oxygen. So this is just essentially saying how much oxygen is around as you go to the right axis. And then the vertical axis tells you how saturated are your hemoglobin molecules. This 100% would mean all of the heme groups on all of the hemoglobin molecules or proteins have bound to oxygen. 0 means that none have. So when you have an environment with very little oxygen, and this actually shows the cooperative binding. So let's say we're just dealing with an environment with very little oxygen. So once a little bit of oxygen binds, then it makes it even more likely that more and more oxygen will bind. As soon as that's why the slope is increasing. I don't want to go into algebra and calculus here, but as you see, we're kind of flattish, and then the slope increases. So as we bind to some oxygen, it makes it more likely that we'll bind to more. And at some point, it just, it's hard for oxygens to bump just right into the right hemoglobin molecules. But you can see that it kind of accelerates right around here. Now, if we have an environment with an acidic environment that has a lot of carbon dioxide, so that the hemoglobin is allosterically inhibited, it's not going to be as good at this. So in an acidic environment, this curve for any level of oxygen partial pressure, or any amount of oxygen, we're going to have less bound hemoglobin. So let me do that in a different color. So then the curve will look like this. The saturation, I'm going to do it darker. The saturation curve will look like this. So this is an environment right here. So this is an acidic environment. Maybe there's some carbon dioxide right here. So the hemoglobin is being allosterically inhibited. So it's more likely to dump the oxygen at this point. So I don't know how exciting you found that, but I find it brilliant, because it really is the simplest way for these things to dump their oxygen where needed. No GPS needed, no robots needed to say, oh, I'm now in the quadriceps and the guy is running, let me dump my oxygen. It just does it naturally, because it's a more acidic environment with more carbon dioxide. It gets inhibited, and then the oxygen gets dumped and ready to use for respiration.
Excuse me. I didn't say. Okay. This is but we will, I guess. Alright, we're gonna read through this down to 83. Down to 83, we'll add Anki cards. We'll review. I think we should also add the kinetic. Might as well add the enzyme kinetic cards to them, and then we'll go ahead and do the other 3. And then we'll finish the rest of the That'll be the day. Blah. Yeah. Oh, I said let's work out, right? All right. Yeah, we will. Let's put 15 minutes on the clock then. Yeah, I need to work out, man. Do something. I need some blood. macro. We're on automatic, right? Yeah, we're on automatic.
Jesus. Jesus.
Sugar and oxygen. You said it's supposed to be blue, but I don't have any more blue. I don't think I have any more. So maybe I should get some this one. Or any of these two. Oh, actually no. This one looks pretty good. Hold on. Excuse me. Velocity with Hmm. Actually, that makes perfect sense. Holy crap. That's beautifully put. Oh my god. That's amazing. Wow. Wow. Vmax, the maximum velocity at which the enzyme can possibly convert substrate to product. Increasing substrate does not increase Vmax, but increasing the enzyme does. Yeah, okay. Gotcha. So if the MCAT practice problem will tell me the enzyme reaches half of its max velocity when the substrate concentration is 2 millimolar. I know that K doesn't have 2 millimolar to reach maximum velocity. We need a substrate concentration at 4 millimolar. Wait. So if I'm using the car analogy, if a bar has a max speed of 100. So VO is max. So max VO is reaction velocity, and that's 100. So we're at 100. It requires 25 gallons to reach half of that. And the substrate concentration requires 25 gallons to reach half of it. 25 gallons to reach the KN. Right. What's its efficiency constant? The efficiency constant is 25. 25. No, I count, I guess, in this case. Gotcha. You added more and more substrate in the cart, and the speed at which the enzyme is turning the substrate to product is increasing, then it flattens out. So 3, that is the V0 max velocity. V0 max velocity, V max. Oh wait, that's not V0. Oops, that's Vmax. Vmax. That's the maximum maximum velocity at which the enzyme can turn the substrates into product. Okay, 4 cars moving at 50 miles per hour. Oh, and the fuel level is at 10. So number 30. I don't think I really need all this. Really, it's the km is 15. Then we know that the v max is going to be equal to 30, and then 3/4 of its max speed. So 22.5 is 3/4 of 30. 22.5. That's 8. Okay, yeah. What's 4? What? Nothing is right. change it? It says 90 now. Oh, it's a censorship. Gotcha. Yeah, me too. Dang it. Dang it, man. What the heck? Wait, so they didn't like that, right? That is actually true. Okay. Oh, I see. Oh, I see. What the heck's going on? Yeah, that's right. Excuse me. Gotcha. Oh, okay, yeah, it's already gonna read. So what is V? Basically, it was on for V. Okay, got it. Oh, okay. Jesus. Let's let's continue reading it, but we'll take it a minute. I think we're almost there, no? I mean, there's only 2 more pages, but we gotta work on it. You know, we gotta practice it to make sure that I got it. Honestly, this is the best. I mean, the sections on mechanical were okay. At least you got a little bit of an idea, but this guide is amazing. I mean, it's so amazing. It's amazing. Amazing, amazo, amazing, amazo. Amazing, amazo. Amazo is amazing. Anyways. What are we talking about here? What are we talking about? What is this button? Yeah. Okay. Where was I? Was there anybody else here? Oh, I don't know what happened. I don't know where all the messages went. Oh, well, whatever. Anyways, let's continue reading it. And as we read it, we go through it. I think it's the best strategy. We gotta get bigger. We gotta get a little more fit too before I go to boot camp. Before I go to boot camp. Yes, sir. Sir, yes, sir. The Lineberg plot used the inverse. I wish I had the actual chart here. What? Oh, did I erased it, I guess. Oopsie daisy. Where is it at? Not you. You. Oh my god. Competitive inhibitor binds to the enzyme. Competitive inhibitor has an increased effect. Binds to the Oh, gotcha. Yeah. Okay, yeah. You'll need to be able to look at a line weaver Burke plot and tell the values of the Km. You also need to know how the intercepts will change as you increase or decrease Km or Vmax. Reversible inhibitors look and how they shift the lines on the intercept. Okay. I should probably spend a little bit more time, but it might be on the card, so let's move on. All right, so I mean that's kind of that, I guess. Now we're on that, so let's cross it out. Let's add the enzyme kinetics card to it, and then do the Anki. Yeah, on the next break, I'm gonna take a quick shower, guys. I don't know. I'm gonna take a quick shower. This guide is the Holy Bible. Take enzyme kinetics on content, read the high yield guide, add enzyme kinetic cards. So basically that, right? So let's go ahead and browse. Let's go to our tape. Let's check this one out real quick. But the rest, do I need this? Know this? I guess so. I don't know. That's pretty much every one of them, I think. Let's just check this one. If not, if this one is true, then yeah, we'll just do it on. We will unsuspend them all. We will unsuspend them all. All right, let's resync it. All right, that's synced. That's synced, and then we still have. Oh, let's try to see if we can answer these questions now, actually. What are you talking about, man? It's negative. Yeah, buddy boy. Doesn't change this. It does not change this. K1. Are you serious? Are you serious? I'm not sure what it was like. Maybe it was one of these. Maybe this one. Sure. It's all invert. Oh man, really? Only working max. KM. Should know that though. But it's not slope. I think it's this one. Oh no. And it's this one. I have to take a look at it again. Buddy boy. Buddy boy. Yeah. S is abundant. ES is at its highest point. Vanishing slowly. This would make sense. ES is vanishing slowly. And this one. But this makes sense too. But it's not really vanishing slowly. It's not really vanishing. Sure, I guess so. It's definitely going to be a low M. I forgot what KCAT was, but I think it's this one. Yeah. So the initial reaction velocity is half of the Vmax. Oh, it's already half of the Vmax, bro. It's 25. You're trying to trick me. You're trying to trick me. Reversible covalent binding of allosteric. Reversible covalent binding of regulatory covalent? Non-covalent. I think it's non-covalent because covalent make it. This one covalent will make it. Covalent will make it irreversible. Increase the activation energy. No. Increase formation of product? Increases the products? What is it? I don't think it's going to increase the products. So it just increases the reaction rate. That's all a catalyst does, increases the reaction rate. Oh, let's go. Boom, baby. So allosteric. It must not be a competitive inhibitor. What is a mix inhibitor? I don't know what that means. Oh, well, mix inhibitor. I don't know what that means. Okay, well, anyway, competitive. Oh my god, what is a mixed? Oh, mix is called non-competitive. Non-competitive mix. I don't know. Pressurizing heat to my. Okay. What are we talking about here? Total protein. I don't know. Very high. Oh. Very low, I guess. Oh, yeah. Okay. I guess, yeah. What else positive? I'm not sure about this one. Yeah, 0.5. Okay, is equal to okay. Oh, okay. Yeah, that's fine. Should we do better on these? I don't know. I don't know if I'd really spend a time. Maybe I probably should. Was that? Let's do the Anki review. And that catalyzes this reaction is an enzyme that catalyzes this reaction is catalyzes transferase. Oh no. I think I think I'm gonna unsuspend this card. I'm sorry, suspend it. This one's fine though. This one is glucose phosphoglucose isomerase. Phosphoglucose isomerase. Bonds and H3 plus H2 don't H2. Oh, C, yeah, C mineral found in arginine. Found in arginine. Found in arginine. Arginine. Yeah, arginine. Catalyst enzymes. The amino serine. Serine sips on a short hydroxyl. Serine ships on a short straw with a hydroxyl tip. Serine sips on a short straw with a hydroxyl tip. Polar because of the hydroxyl group is involved in the attack of phosphate groups because the hydroxyl group serine, tyrosine, and 309. Exergonic reactions require release energy. The release energy exergonic reactions occur when the was it free energy? When it gives free energy is less than 0. Right, but essentially isn't this the energy released? Negative delta G, negative delta G. What was this called again? Gives free energy change. Formation of products. Reactant products. Yeah. And then the reaction is not spontaneous. Reactant. Oh, this is on the reversal side, right or no? Energy is added. Reaction is non-spontaneous. 309. 309 is 309. Yeah, 309 is polar. Polar is a short tree with a methyl and a hydroxyl group. Polar uncharged, polar neutral. Require energy. And it is positive delta G. Positive gives free energy. Amino acid glutamate. Glutamate. Glutamate is we got some food. Let's go. Let's go. Is acidic, negatively charged glutamate. Longer chain than aspartate by 1 carbon. Let's go. They release negative delta G. Negative free gives energy, which is the amount of energy it requires to go from reactants to products. And then reacting to the transition state is activation energy, and that's how we use the catalyst to reduce that activation energy. Oxidoreductase. Nonpolar aromatic has the indole group. It makes for less than light. Transferase. It's found in histidine, cyclical structure without nitrogen. 2 double bonds. No, 2 nitrogens. Hydrolyze. LNS. Chiral. Lyase. But does it with the formation of double bonds and ring structures. Isomerase. Enzyme. Polar. Hydrophilic. Yeah, polar hydrophilic. Short chain. And neat amide. Neat amide. Oh, isomerase. Phenylalanine. Hydrophobic. And there's a benzyl group after the carbon on the R chain. Aromatic. No charge. Absorbs UV light. Release. Oh, well. And the organic require, positive. Delta 2. I missed that. Isomerase. Also, glucose isomerase. release oxidoreductase. Ligase. Transferase. Kinase. Transferase is a kinase. Require positive delta G. Glutamine, the quinidine, longer chain, 2 carbons, and longer chain bicarbonate, and contains amine group as well. Q, polar, uncharged aliphatic. Removes the phosphate group. Removes the phosphate group? Which one of these removes the phosphate group? That's a good question. Yeah, I'm not too sure. Oh, phosphatase. Phosphatase. Which one is that under? I was gonna say hydrolase, yeah. Okay. Tryptophan, a W, tryptophan W emits light, contains the indole group, just like there's an aromatic. I'm gonna say polar. Oh, whoops, it's non-polar. This version like non-polar. Transferase kinase. Proline, non-polar, hydrophobic. Proline forms the cyclic ring structure. Disrupts alpha helixes and is found in the beta turns. As causes kinks. Yeah. Folds onto itself. Hydrophobic. Is an amino acid, yeah. Phosphatase, which is under hydrolase. Transferase. Hydrolase. Like phosphatase. I just did this one. Kinase transferase. Lyase. Getting rid of a bond. Is shaped like that. Hyperbolic. Aspartate, negatively charged, double O's, shorter than glutamate, which is E. The KM. KM is here. Cysteine polar. Cis, C. Cysteine brushes her hair with a sulfur handle. It's going to end with a SH group. Oh, with a short sulfur handle. Cysteine brushes her hair with a short sulfur handle. It's shaped like a hyperbolic. No, hyperbolic is hyperbolic. Bmax is when all the enzyme's active sites are saturated with the substrate. Yeah, it's lysine, pola, lysine, long chi, the NH3 plus at the end. It's V0 is equal to Vmax. Vmax times concentration of substrate divided by Km plus the concentration of the substrate. Yeah. Asparagine, polar, asparagine, and ESN, polar, short neat amide. short neodymium. Phenylalanine absorbs UV light, contains the benzyl group, hydrophobic. No. Hydrophobic. Aromatic. Hydrophobic. Absorbs UV light. Yeah. Histidine contains the imidazole group, and it's polar, positive charge, basic. Yeah. Oh, neutral physical pH, which, yeah, pKa 6. I'm not sure. Sigmoidal. Oh, that's right. Ligase. Summaries. Basic, positively charged. Arginine. Contains a short neat amine. No. R. Wait, arginine. Wait. Arginine. Oh man, arginine. Arginine. Oh, contains the quinidinium group. R. Rocket with a quinidinium group. Wait, yeah, so this one's, yeah, basic, very basic. Rocket with a quinidinium group. Yeah, quinidinium group, yeah. Cooperative enzymes display a sigmoidal shape curve because of the activity in the initial substrate binding. And they're required and have a positive delta G. Tryptophan contains the indole group, absorbs UV light, W, aromatic, yeah, hydrophobic. Changes the binding affinity, cooperative ability, cooperative binding, yeah, cooperative binding. Tyrosine is like phenylalanine but contains a hydroxyl group. It is grouped with serine, tyrosine, 309, hydroxyl groups. Y Y aromatic. Yeah, aromatic polar. Active site. The indole group. Two ends and double bond. Oh no, the indole group. Yeah. Benzene and cyclical ends found in tryptophan. Remove hydrolase phosphatase. What is this one? The induced model fit? Induced fit model? Induced fit theory. That the enzyme and the substrate both undergo conformational changes. Transferase, kinase, KM, hyperbolic, maximum rate at which an enzyme can catalyze a reaction, Vmax, is hyperbolic. Oh no, whoops. Sigmoidal. V0 is equal to Vmax times concentration of substrate divided by Kn plus the concentration of substrate. Cooperative binding, active site. This is the indole group found in tryptophan, benzene, and a cyclic nitrogen structure. And then induced fit theory. V0 is equal to Vmax times concentration of substrate divided by Km plus concentration of substrates. Okay, well, let's take a 10-minute here. Man, I need to do at least one more today, dude. I'm supposed to be on gene control. I mean, we still have time. Maybe I can stop at 10. So, about an hour. I think we might still be able to do it. It's just going to be like a quick run through. But we'll see. I don't know. Okay, what was I? Oh, yeah, I was going to take a 10-minute. Jesus. We got 2 more decks, and then we can go to lesson 5. DNA structures and functions. Read through that and continue. Let's share the deck. If we can get through this today and have oh man, I'd be so happy. What is this one? Okay. Let's continue. tRNA. Amino acyl tRNA synthetase. Different synthetase for each amino acid. I mean, I'm not here yet. Oh, never mind. I wonder where this one's at though. Ligase. H3 plus. C double bond A minus hydrogen group and an R group. Oh, CO. Protonated neutral. To break the bond, we add water. This is called condensation reaction. No. Dehydration. Oh, it's called. Hold on. Breaking the peptide bond requires them to break the bond. We add water. It's called dehydration reaction, hydrolysis, or even dehydration reaction. Molecules used in the process. Hydrolysis reaction. Ligase using ATP. I should do it in the morning, but I needed to finish this one and then have more for tomorrow morning. We're talking about when we think we're talking about speeding up the reaction. Speeding up the reaction. We're talking about flavor. And that kinetic studies how quickly, the rate. Oh my god, the rate. Speed of reaction rate. Yeah, rate. Oh god. Proteins below the amino acid below proteins conform to bonds. In terms of many things, but its true application is as a saturation curve that shows the relationship between the saturation concentration the MM graph. Okay, MM kinetics. Okay, let's maintain kinetics. It's a combination of both ionic ionic. Yeah. Ionic and hydrogen bonding. Ionic and hydrogen bonding. The rate glutamic acid. Glutamate. Enzyme impact reaction rate, not equilibrium. And they also say electric transition state. That's true. Impacts kinetics. I think it's the same thing. Reaction rate kinetics. I think I'll be good for that. To calculate the isoelectric point, identify the ionizable groups. Identify the ionizable groups and calculate the average. Identify the ionizable groups. And then calculate the average. The average of the two central pKa values, where pKa1 is the carboxyl group, pKa2 is the amino group, pKa3 is the R group. Let's go. And then two central pKas, always pick above and below the 0. Representation of the enzyme kinetics, where S is the substrate, E is the enzyme abusive product. We have E plus S turns into ES turns into E plus P. Disulfide bonds. Disulfide bonds? Disulfide bonds? Like, I guess like serine, right? Disulfide bonds like serine amino acids. Not serine. Cysteine. I meant cysteine. Cysteine has a short handle. Cysteine has a short sulfur handle. Heat can disrupt these bonds. Two cysteine residues. Yeah. Length is a group. Changes between them. The R group. Coenzymes often serve as carriers of electrons. Organic molecules. Specific functional groups or electrons. Okay. Every protein has a sarcomere AUG. 3 codons that act like stopping signs. UAA. UAG. UGA. Yeah. Amino acids. Cofactors are minerals, magnesium plus, and others, whatever, that help to facilitate the enzyme catalyzer reaction. Proper functioning of some enzymes, yeah. Yeah, inorganic ions. Inorganic ions. Yeah. Adenine, thymine, guanine, cytosine. Thymine is replaced by uracil. Proline known for breaking up helixes and being in beta turns due to disrupting the rotation in the alpha helix. Yeah, disrupting the rotation in the alpha helix. Kinetics of reaction do not alter equilibrium. They do not alter the free Gibbs energy, delta G. Oh, they do not alter the thermodynamics. Proteins are phosphorylated to. Proteins are phosphorylated to. Can be phosphorylated include serine, tyrosine 309, known as the phosphomimetic effect. Commonly phosphorylated to. I don't know. Oh, that's right. Upregulate or downregulate their function. How fast the protein is synthesized. This can be sped up with enzymes within a cell. It's called chaperone. A chaperone. Well, without sticking to other proteins, and they can even help fix mistakes by pulling back folds and glucose. It's crucial because the person taking them together fast is fast chaperone protein. Yeah, that's crazy. Enzymes, while influencing the kinetics reaction, do not all alter thermodynamics. Charges of the essential amino acid can carry charges determined by determined by the R group. The essentials amino acid and the essentials. Oh yeah, milk. Milk for the win. Milk for the win. Yeah, milk for the win. Amino acids carry charges determined by the ionization of the functional groups in response to the environment, which are called codons, which are called mutations. These change the codons in the DNA. Ligase. Between the rate of the reaction and the substrate concentration. Yeah. Is the is due to the oppositely charged the functional groups of the oppositely charged amino acids to bind together in ionic bonding and hydrogen bonding. Yeah, it's basically it's determined by many things. The Michaelis-Mendelian Michaelis-Mendelian graph, Michaelis-Mendelian model, Michaelis-Menten kinetics, Michaelis-Menten kinetics. Enzyme kinetics do not affect thermodynamics. And they don't change the delta G. But the destabilized transition state not affect equilibrium. At which it carries no net charge. And the net charge of an amino acid is the net charge of an amino acid is the pseudorad. Wait, the net charge of an amino acid is the summation of all the ionizable groups. Yeah, it's a summation of all the ionizable groups given that are given in response to that are determined in response to the environment of the amino acid in various environments, such as like pH and temperature. Right? Oh yes, okay, yeah. Some charges. Yeah. We're here to work kinetics with two phase sync movement, and we're discussing the kinetics. We're talking about the rate. E is the enzyme concentration. These codons tell so codon AUG. Speed up the reaction. Individual amino acids undergo a condensation reaction, which are held together by covalent bonds, peptide bond. S. E plus S is ES, E plus P. Impact reaction rates. Kinetics. You would expect only hydrophobic amino acids like glycine, leucine, isoleucine, alanine, methionine, valine, isoleucine, proline, and phenylalanine F. Carriers of coenzymes are carriers of molecules and electrons. Well, specific functional groups or electrons. Or electrons. They come together and cause folds. Alzheimer's disease. Oh, cause diseases. It's formed between the amino group of one amino acid, which acts as a nucleophile, and the carboxyl group of the other amino acid trying to add, which acts as the electrophile. Amino group nitrogen, carbonyl carbon. carriers of functional groups or electrons. The Michaelis-Menten equation causes diseases or causes Alzheimer's. Identify the ionizable groups, determine the pK values. Amino, A carboxyl, 3. Histidine's pK is 6, which is neutral at physiological pH. Aspartic and glutamic acid are 4, and lysine and arginine are 11 and 12. Inorganic are ions or inorganic molecules. Oh, inorganic ions or small molecules. In an oxide with a negatively charged substrate, you choose medium charged substrate. You choose a positive amino acid like histidine, lysine, or histidine, lysine, arginine. And then kinetics. Thermodynamics. Are inorganic ions or small molecules. Upregulate or downregulate their functions. Upregulate or downregulate their functions. Tyrosine 309 and serine. Phosphoramimetic effect. Fast. Chaperone protein. Determined by their functional groups in that is determined in response to their environment, such as pH and temperature. The ionization of the functional groups. In response to pH, but if it's ionization, it's just pH. between the reaction rate and the concentration of the substrate. This implies the transition state. The isoelectric point is refers to gel electrophoresis when the net charge to identify the pH value at which the net charge of the molecule of protein amino acid is 0. List them in order and choose the pKa's that are above and below the net charge. And P plus P have a N terminal and a C terminal with the first message are amide groups. Enzyme impacts kinetics, not thermodynamics. Equilibrium. E. Not equilibrium or thermodynamics. Speed up the reaction. Diseases. Alzheimer's disease. Organic small molecules. Or electrons. Specific functional groups, organic small molecules, or electrons. Michaelis-Menten equation. Destabilizer transition states. Cofactors or inorganic ions or small molecules. PP. Not equilibrium or thermodynamics. IFD. 6 new 22. Salt bridge stabilized tertiary structures. Six. Neutral physiological pH. 4. Kinetic. Catalysis is a kinetic parameter. Le Chatelier's. 50%. Covalent bonds. Peptide bonds. Um, 12. Enzymes cannot Do not. Hydrogen group. pKa. Carboxyl group, COOH, carboxylic acid. Kinetic parameter. The rate of the reaction. 3. R group. Serine 309 tyrosine. Anticodons. 11. pKa. Undergo a hydrolysis. Oh. No. Condensation reaction. We're not cleaving the bond, we're giving off water. Not using water to cleave the bond. Okay. During translation. Oh, elongation. Is elongation in translation? I think so, right? Elongation. I think it's the last step of translation. Occurs in the ribosome. Amino group. Threonine, tyrosine, and serine. Eight. Concentration. Equilibrium. Hydrogens. Serine, threonine, tyrosine. Elongation. And that occurs in the ribosome. Equilibrium. Rate of the reaction. Do not. Kinetic parameter. Anticodons. condensation reaction. Elongation. Ah. Equilibrium. Okay. I don't know how fast I can really go through this, but I can try. Luckily, there's not that many articles. There's like 3. What about the next one? The next one, it's the same thing, only 3. The next one for that, there's only 1. Oh wow. We might still be able to make it. We'll see. If we do make it, then I'll add the Yankee cards and I'll study all of them again tomorrow morning. Oh, that'd be so nice. Let's see. We still got time. We can make it. I have to go back to now. Oh man, I need parts on. I need to put on some a jacket or something. I do want to take a shower though. Yeah, I'll probably just continue. I guess. Anyways, we'll see. For now, let's put on some socks. Maybe on the next break weekend. Maybe on the next break weekend. We can work out again. Right, yeah. Let's go. Well, I did the Yankee. I still, probably need to go back. We'll see. gene control. I mean, transcription, transmission. I kind of went over it a little bit already. But I think I could do it pretty fast. Anyways, let's continue. For now, let's put on the hold on. Okay, yeah. Okay, let's continue. And we got these two. We need to end that. Okay, then we can finish. Maybe what does it say? A little something. Let's put on the jacket, huh? These are one. Oh, my foot. My foot fell asleep. Kind of just my toe. Ow. Sure. Let's go. Let's go. Deoxyribose. Deoxyribose. DNA. Urine contains one oxygen. Deoxyribose looks like this, mate. One oxygen and 4 carbons. A 5th carbon atom is attached to the 4th carbon of the ring. A 5th carbon is attached. Anyway. A 5th carbon atom is attached to the 4th carbon ring. 5th carbon atom is attached to the 4th carbon ring. Can we take a look at that real quick? Deoxyribose. Where are we talking about? Where are we talking about? What are you talking about, mate? What are you talking about, mate? Talking about right here. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Let's go. Attached to the 4th carbon ring right here. Hydroxyl group attached to the 3rd carbon in the ring. What? What are you talking about? What are you talking about? Was it really? 3rd carbon ring is here. Oh, yeah, that's true. You're right. You're right. I would hope so. And nitrogenous base. In this example, this is adenine. And this is the phosphate group over here. P O O H NO minus and NOH here. Lovely. 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 Ta. Phosphate group is a phosphorus atom. Phosphorus atom with 4 oxygen atoms bonded to it. No, it's going red. The oxygen sticking out of the oxoribose sugar of another nucleotide? What are you talking about? Phosphorus atom in the thought. Here. I'm talking about here. The hydroxyl group. Is that what you're talking about? Mate. Let's take a look at the structure. Will you? Take a look at the structure. Will you? Will you? Take a look at the structure. Will you? Can somebody give me a good visual? You? Is it you? Is it you? Is it you? Wait, where'd you go? Oh, come on, that was a good one. Come on, man. Come on, man. Nah, seriously? Right there. Oh, it's not. It doesn't work. Yes, sir. That's a good one right there. That's a good image. Yes, sir. Yes, sir. Yes, sir. I see it right here. Yes, sir. You're absolutely right. You're absolutely right. Contains an adenine. Put you right there. You contain a thymine. Put you next to the adenine. We're going to image you. Close you. Image you. Close you. There's my adenine. Adenine. Thymine. We're going to image you. Put you right there. Put you right there. And my cytosine. And my cytosine. Give me a good image. There you go. There you are. Wait, next to this guy. Adenine. Guanine. Thymine. Okay, you got oxygen there. You got oxygen there as well. But you're a double cyclic structure. You're a double cyclic structure. You're a single structure. And you're a single structure. Single structure, double structures. Okay. Multiple nitrogen atoms. Man, how long is this? Okay. Let's put you out of here. Actually, I want to keep it because I want to wait until it's full. Nucleotide monomers and DNA polymer. Excuse me.